Hello and welcome back to the Forgotten Deep. Today, let's relax and wind down, in a place where curiosity is allowed and sleeping is preferred. Now let us begin learning about the ecosystem found beneath Antarctic ice. Imagine standing at the edge of the world. Not metaphorically, literally. You are on the deck of a research vessel in the Bellingshausen Sea, on the western side of Antarctica's peninsula, and everywhere you look, there is nothing but ice, ocean, and a sky so vast and so cold it feels like the universe itself forgot to put a ceiling on it. The water here is a shade of blue that doesn't have a proper name yet. Not quite navy, not quite gray, something between midnight and slate that shifts depending on whether the clouds part or close. The temperature on deck sits well below freezing. The wind doesn't blow here so much as it insists. Every surface on the ship is dusted with salt and the memory of spray. You are about as far from civilization as a human being can get and still be on this planet. Now imagine that somewhere out there, right now, in January of 2025, the ice is breaking. Not cracking, not splitting, breaking. In a single catastrophic, geological event that has been building for years in the dark. A slab of ice roughly the size of the city of Chicago tears itself away from the Antarctic mainland and begins its slow, grinding drift into the Southern Ocean. Scientists will give it a name, the way they give all Antarctic icebergs names. A84. A bureaucratic designation for something that is, by any reasonable standard, one of the most staggering physical events a human being can witness. 30 kilometers long, 17 kilometers wide, 150 meters thick, meaning that if you dropped the Statue of Liberty down through its center and kept stacking copies of it end over end, you still wouldn't reach the bottom. Hey, 84 weighs hundreds of billions of tons, and it is moving, slowly but unstoppably, into open water, and where it once sat, where it pressed down into the Bellingshausen Sea for decades, perhaps for centuries, there is now nothing but open ocean. And waiting on that open ocean, by a coincidence so perfect it sounds scripted, is the research vessel Falker, too. Here is where the story gets interesting. The scientists aboard the Falker had not come to Antarctica to find anything hidden under the ice. They were there to study the seafloor geology at the margins of ice shelves, to understand how these ancient, frozen structures had changed over time, how they'd shifted and retreated and grown in the long geological memory of the planet. It was a serious, methodical expedition, the kind of science that doesn't make headlines very often, the kind that involves sediment cores and acoustic mapping and years of patient data analysis. They were not treasure hunters. They were not expecting anything to surprise them. And then A84 broke off. The scientists on board had 12 days to make a decision. 12 days before the currents and the winds pushed the iceberg far enough away that the newly exposed seafloor, 510 square kilometers of it, a stretch of ocean bottom that had never once been touched by sunlight, never surveyed by humans, never accessed by any instrument in the history of scientific exploration, would start to change. 12 days before whatever was down there in the dark, sealed under centuries of ice, became just another unremarkable stretch of the Southern Ocean floor. They didn't hesitate. Dr. Jyotika Vermani, the executive director of described the moment they made the call to change course as a serendipitous moment. Expedition co-chief scientist Dr. Patricia Esquite, a marine biologist from the University of Aveiro in Portugal, put it more directly. We seized upon the moment, changed our expedition plan, and went for it so we could look at what was happening in the depths below. What they found would rewrite what we thought we knew about life on Earth. Beneath the ice, in darkness so complete that no photon of sunlight had penetrated it in possibly hundreds of years, in water so cold it hovers just above the freezing point of salt water, at depths reaching down to 1,300 meters below the surface, something was alive. Many things were alive, not just surviving, not just clinging to existence in the desperate, minimal way you might expect from organisms trapped in one of the harshest environments on the planet. They were thriving. Corals and sponges that took decades, possibly centuries, to grow to their current size. Octopuses resting on the seafloor a kilometer down. giant sea spiders, icefish, a phantom jellyfish, one of the rarest animals in the ocean, drifting through the dark like a ghost. An ecosystem so complex, so fully realized, so deeply alive, that the scientists watching the ROV footage in the control room above could barely believe what they were seeing. We didn't expect to find such a beautiful, thriving ecosystem, Skeet would later say. Based on the size of the animals, the communities we observed have been there for decades, maybe even hundreds of years. Hundreds of years, under the ice, in the dark. This is a story about what happens when the planet surprises us, about what life is capable of when we are not watching, about what hides beneath the frozen skin of the most remote, the most hostile, the most inaccessible place on Earth, and what the discovery of it means for our understanding of life itself, not just here, but potentially on other worlds. This is the story of the ecosystem found beneath Antarctic ice. Section 2. The discovery. To understand how remarkable the discovery of A84's hidden world truly was, you first need to understand just how extraordinary it is to even be in the Bellingshausen Sea in the first place. Antarctica is not a single thing. People who have never studied it tend to think of it as a kind of uniform frozen wasteland, a white expanse, uniformly hostile, uniformly remote, uniformly unknowable. But Antarctica is a continent of regions, and each region has its own character, its own peculiar combination of geography and oceanography and climate. The Bellingshausen Sea, which sits on the western side of the Antarctic Peninsula, the long finger of land that reaches northward towards South America, is one of the most dynamic and least understood of all Antarctic regions. Its sea ice is seasonal, advancing and retreating dramatically with the southern hemisphere's summer and winter. Its waters are among the most rapidly warming in the entire Southern Ocean. And perched along its coast, covering a total area of roughly 25,000 square kilometers, is one of the most significant and most vulnerable ice formations on the entire continent, the George VI Ice Shelf. The George VI Ice Shelf is not a glacier. That distinction matters and is worth pausing on. A glacier is ice that flows down from land toward the sea. An ice shelf is what happens when that ice reaches the ocean and keeps going. It floats out over the water, attached to the land at one end, suspended over the deep ocean at the other. The George VI Ice Shelf sits within a long, narrow body of water called George VI Sound, which separates Alexander Island to the west from Palmer Land, part of the Antarctic Peninsula, to the east. It is, along its center line, approximately 450 kilometers long. In the north, it is about 20 kilometers wide and roughly 100 meters thick. In the center and south, it balloons out to 60 or 75 kilometers wide and thickens to as much as 600 meters. At its southern end, it opens into the Ron entrance, the bay where A84 would eventually calve. The ice shelf was first glimpsed from the air in 1935 during a historic trans-Antarctic flight by the American explorer Lincoln Ellsworth. A year later, the British Graham Land expedition under John Riddoch Rymil explored the region more systematically. It was eventually named in honor of King George VI of England, a naming that was only formally confirmed and recognized in 1975 through a joint determination by both the American Advisory Committee on Antarctic Names and the British Antarctic Place Names Committee. For the better part of a century since its discovery, the ice shelf has been studied, mapped, debated, and watched with increasing urgency by glaciologists who understand that it sits, according to some models, very close to its thermal limit of stability. It has retreated at both its northern and southern fronts in recent decades. It has thinned. In places, it has accelerated. Scientists have looked at its history in lake sediment cores from the islands around it and found evidence that the ice shelf has, at least once in the distant past, around 6,500 years ago, retreated so far that open marine conditions existed in George VI Sound. They found fossil barnacle shells from that era, creatures that could only have lived there if the ice was gone. The ice shelf came back. It always has. But the question that haunts researchers today is, will it come back again? All of that history, all of that context, is the backdrop against which the story of A-84 unfolds. By late 2024, satellite imagery had already revealed a significant rift developing in the George VI ice shelf's southern end, a fracture in the ice that had been extending and deepening as the shelf's internal stresses built up. But for a while, Iding was hemmed in by seasonal sea ice at the Ron entrance. Think of it like a car parked behind a snow bank. The machine is ready to move, the engine running, but the path is blocked. As the Southern Hemisphere's summer advanced into January 2025, that seasonal sea ice began to melt back, opening the way. On January 13th, 2025, the crack reached its terminus. The ice shelf fractured completely along the rift line, an A-84, 30 kilometers long, 17 kilometers wide, potato-shaped according to NASA Earth Observatory imagery, and thick enough to dwarf most skyscrapers, separated from the mainland of ice and began to drift. It moved fast, faster, in fact, than glaciologists expected. Christopher Schumann, a retired glaciologist from the University of Maryland, Baltimore County, monitored A-84's progress via NASA satellite imagery and was caught off guard by its speed. Carried by Antarctic coastal currents, the bird ricocheted along the coastline, traveling some 250 kilometers in its first month. "I'm impressed by how fast it has moved in the coastal current," Schumann told NASA's Earth Observatory. And then he added a line that perfectly captures the scientific hunger that drives exploration at the edges of the known world. "It makes me wonder just what is happening in the water under the ice shelf." It turns out Schumann wasn't the only one wondering. The R/V Falker, too, was already in the Bellingshausen Sea when A-84 calved. It is worth taking a moment to appreciate just how remarkable a machine this ship is, because it is not simply a boat with some scientific equipment bolted to it. The Falker, too, named, whimsically, after the luck dragon from the German fantasy novel The Never-Ending Story, continuing the tradition of the first R/V Falker, is a 110-meter, 363-foot state-of-the-art oceanographic research vessel that was originally built in 2011 as an offshore industry ship, later used to help establish wind farms in the North Sea, and then purchased from a Norwegian shipping company in 2021 and completely refitted into one of the most sophisticated mobile scientific laboratories on Earth. It has 8 laboratories, 3 multi-beam echo sounder arrays, 7 over-the-side launch and recovery systems for scientific equipment, 70 berths, one of the largest cranes ever mounted on a research vessel. Its aft deck alone covers nearly 1,000 square meters of working space. And at the heart of its scientific capability, the thing that makes the Falker, too, not just a ship but a portal into the deep ocean, is ROV Sebastian. Sebastian, another never-ending story reference, named after the human boy at the center of the novel, is a remotely operated vehicle that represents the cutting edge of deep-sea exploration technology. It weighs 3,200 kilograms, roughly the same as a large SUV or a small adult elephant. It measures about 2.7 meters long and 1.8 meters on each of its other dimensions, roughly the size of a subcompact car, though far more densely instrumented. It is fitted with syntactic foam, a material specifically engineered to provide buoyancy under extreme pressure, allowing Sebastian to remain neutrally buoyant as it descends, neither sinking nor floating, hovering at whatever depth its pilots direct it toward. 5 thrusters control its movement, 2 for forward and backward, 2 for vertical, and 1 for lateral sliding. It is tethered to the ship above by an umbilical cable that carries electrical power down and data back up, a sophisticated lifeline using a cantilever system specifically designed to prevent the cable from fouling in the ROV's own propellers. It can reach depths of 4,500 meters, well over 2.5 miles, and it carries a suite of cameras, lights, manipulator arms, and scientific sampling equipment that allows scientists aboard the ship to see, measure, and collect from environments that no human being could ever physically survive. When Sebastian dives and transmits its footage back to the ship, the entire scientific team can watch in real time in the control room, a space with 4 captains' chairs and over 20 monitors, customizable to display multiple simultaneous video feeds, sonar data, depth readings, and navigation information all at once. And crucially, all of those dives are live-streamed to the public. That has a policy of radical transparency. If Sebastian is in the water, the world can watch. Now, the science team aboard the Falcor, 2, in January 2025 had not come to the Bellingshausen Sea to look under the George VI ice shelf. They were there on an expedition that was officially part of Challenger 150, a global cooperative for deep-sea biological research endorsed by the Intergovernmental Oceanographic Commission of UNESCO as part of the International Ocean Decade Program. The original expedition plan was to study the seafloor and ecosystem at the interface between ice and sea, the zone where glacial dynamics and marine biology intersect in complex and still poorly understood ways. The team was led by two co-chief scientists, Dr. Patricia Esquite, the marine biologist from the University of Aveiro in Portugal, and Dr. Sasha Montelli, a geophysicist and glaciologist from University College London, also a 2019 Schmidt Science Fellow, a prestigious award given to early career scientists for cross-disciplinary thinking. The international team also included researchers from Chile, Germany, Norway, New Zealand, and the United States. They were doing good science, important science, the kind of science that builds the patient, detailed, brick-by-brick understanding of how Antarctica works that scientists will need to make reliable predictions about its future behavior. And then, 12 days after A84 calved, they arrived at the site where it had been. On January 25, 2025, the R/V Falcor 2 reached the newly exposed seafloor, 510 square kilometers of ocean bottom that had been hidden beneath more than 150 meters of floating ice for an unknown, but certainly very long period of time. They were the first human beings in history to be in a position to investigate it. No ship had ever been there. No ROV had ever dived in those waters. No satellite could see through the ice that had covered it. Whatever was down there had existed in total isolation, in total darkness, unknown and unseen, until this moment. Sebastian went in. The descent takes time. Even at its operational speeds, dropping from the surface to depths of several hundred and eventually over 1,000 meters is not instantaneous. There is a long period, watching the monitors as the blue of the shallows gives way to something darker and darker, the surface light fading, the world above receding into a memory, the pressure outside the ROV's hull increasing with every meter of depth in a way that would crush an unprotected human being in moments. The ROV's lights cut small cones of illumination in water that is otherwise perfectly, completely black. For a while, there is nothing to see but those cones of light and the particles of marine snow, the constant microscopic rain of organic debris that falls through the water column in every ocean on Earth, drifting silently through the beam. And then, the seafloor. The first image that resolved on the monitors was not what anyone expected. It was not a barren, scoured, lifeless plain of sediment, which is what the conventional scientific wisdom about sub-ice shelf environments would have predicted. The floor of the Bellingshausen Sea, in that long-hidden space where A84 had once been, was covered in life. A massive sponge, larger than a man could wrap his arms around, sat at the center of the frame. Perched on top of it, apparently completely unbothered by the arrival of a 3,200-kilogram robotic vehicle with powerful lights pointed directly at it, was a crab. The scientists in the control room, watching on the monitors, stared. And then, over the next 8 days, 8 days of diving, 8 days of Sebastian dropping into the dark and sweeping its lights across a world that had never been seen before, the full scale of what had been hiding under the ice began to reveal itself. Sponges. Enormous ones, pale and intricate, their branching structures reaching upward in the way that sessile filter feeders reach, blindly, toward the flow of whatever nutrients the water currents bring. Sea anemones clustering around the base of rocky outcroppings. Corals, cup corals, cold water corals, filter feeding colonies built up over what must have been extraordinary spans of time. snails moving across the sediment with the unhurried patience of animals that have never been disturbed in their evolutionary memory. Worms. Sea spiders, not the fragile, spindly creatures you might picture from a tide pool, but giant sea spiders, the Antarctic variety, pycnogonids that can grow to extraordinary sizes in the cold, oxygen-rich waters of the deep Southern Ocean, a phenomenon known as polar gigantism. Ice fish, long-bodied, pale, translucent blooded predators that are among the only vertebrates on Earth whose blood contains no hemoglobin at all, surviving in these conditions through a unique and still fascinating biochemical adaptation. And deeper, in the underwater ravines and canyons that cut through the shelf slope like geological scars, at depths reaching 1,150 and then 1,300 meters below the surface. An octopus, resting on the seafloor with the boneless, liquid ease of a creature perfectly adapted to its environment. And in the water column above, not on the bottom but drifting, translucent, enormous, the unmistakable form of a giant phantom jellyfish. The giant phantom jellyfish, Stygiomedusa gigantea, is one of the rarest animals in the ocean. In all of recorded marine science history, it has been encountered fewer than 130 times. It can grow to a meter in bell diameter, with ribbon-like oral arms that stream behind it for more than 10 meters, making it one of the largest invertebrates on Earth by total body length. It has no tentacles. It captures prey, small fish, crustaceans, by engulfing them in those trailing arms. It lives in the mesopelagic and bathypelagic zones, the so-called twilight and midnight zones of the ocean, typically at depths between 200 and 1,000 meters. It is so rarely seen that a significant portion of everything science knows about it comes from footage captured incidentally by ROVs on expeditions where no one was looking for it. To encounter one, on the first expedition ever to reach this particular stretch of seafloor, in a place that was covered in ice 12 days before, was the kind of moment that marine biologists describe in ways that tend to run out of adjectives. The team used Sue Bastian's manipulator arms to carefully collect samples, biological specimens, sediment cores, water samples. Among the collections, expedition scientists identified what appeared to be a new species of isopod, a crustacean related to the familiar pill bug, though the deep-sea Antarctic variety is considerably less familiar and considerably more alien in appearance. Dr. Eskeet is photographed on the ship's deck holding the small, segmented creature in a sample jar, peering at it with the concentrated expression of a scientist who knows she may be looking at an animal no human has ever named. We will undoubtedly make new discoveries as we continue to analyze this vital data, said Dr. Montelli after the expedition. The word undoubtedly is significant. Not hopefully, not possibly, undoubtedly. The samples collected during those 8 days of diving will take years to fully analyze. Genetic sequencing, morphological description, biochemical analysis. The scientific papers that emerge from the expedition will continue to appear for years, perhaps decades. But beyond the individual species, beyond even the beauty and strangeness of everything Sue Bastian's cameras captured, the discovery raised a question so fundamental, so deeply challenging to our existing understanding of how life works at the bottom of the sea, that it transcended the biology itself and entered the territory of genuine scientific mystery. How is any of this possible? The seafloor ecosystems that science has described in detail, the ones we have models for, the ones we have theories about, depend, at their most fundamental level, on energy from above. In the sunlit surface ocean, phytoplankton photosynthesize, converting sunlight and carbon dioxide and nutrients into organic matter. That organic matter gets eaten. The things that eat it get eaten. Eventually, through a cascade of consumption and death and decomposition, some fraction of that original solar energy makes its way to the seafloor in the form of marine snow, the constant downward drift of organic particles that Sue Bastian's lights illuminate during every deep dive. Deep-sea ecosystems are, in essence, the beneficiaries of a slow-motion energy rain that originates at the surface. But the seafloor beneath 84 had been sealed under 150 meters of solid ice for an unknown but potentially very long time. Possibly decades, possibly centuries. The ice above it blocked the surface ocean entirely. No photosynthesis was occurring in those waters because no sunlight was reaching them. No marine snow was falling through because there was no productive surface ocean above to generate it. The conventional supply chain of energy, sun to phytoplankton to marine snow to seafloor, had been completely cut off. And yet the ecosystem was not just present. It was not just surviving. The sponges were enormous, and sponges grow less than 2 centimeters per year in Antarctic waters, meaning that some of the specimens Sebastian observed must have been alive for decades. The corals were structurally complex in ways that take time, years, decades, to develop. The entire community had, in the words of Dr. Eskeet, been there for decades, maybe even hundreds of years. So how? How does a community like this sustain itself without sunlight, without marine snow from a productive surface, without any of the energy sources that deep-sea ecology textbooks say it should need? The scientists aboard the Falker, too, deployed additional instruments before they left the area, autonomous underwater vehicles called gliders, which move through the water column taking continuous measurements of temperature, salinity, current speed and direction, and chemical properties. Preliminary data from those gliders suggested something intriguing. There was high biological productivity in the region and a surprisingly strong flow of glacial meltwater from the George VI ice shelf itself. The meltwater, fresh water, less dense than salt water, flowing upward from the base of the melting ice, was mixing with the surrounding ocean in ways that could, conceivably, drive circulation patterns capable of transporting organic material from distant productive surface waters into the dark space beneath the shelf. Could it be currents? Ocean circulation carrying food from distant places, threading it under the ice shelf along the seafloor like a secret supply line? Could it be chemical energy, chemosynthesis, the same metabolic strategy used by the organisms that live around hydrothermal vents, which harness the energy in chemical reactions rather than sunlight? Could it be the meltwater itself, carrying dissolved organic carbon from the melting ice above? Or is it something else entirely? Something that the expedition's instruments were not specifically designed to measure, a mechanism that science has not yet formalized? These questions were not answered on the Falker 2. They could not be. The expedition had pivoted from its original mission on 12 days' notice. It had done extraordinary science in a very short window of time, but it had not arrived equipped to solve every mystery it uncovered. The data collected, the samples, the footage, the glider measurements, the sediment cores, will take years to fully process. The mystery is real, and it is open. Dr. Jyotika Vermani, the executive director of the, said it plainly. This is all very much an area of active scientific research now because of this discovery. And Joan Bernhard, a biologist from the Woods Hole Oceanographic Institution who reviewed the findings, offered a measured but striking assessment. Such abundance implies currents are strong enough to transport sufficient organic matter to this area. The key word there is implies, not proves, not confirms, implies. Science is honest about what it knows and what it doesn't, and what the January 2025 expedition established beyond any doubt is this. Something is feeding the ecosystem beneath the George VI ice shelf, and we don't yet know exactly what it is. What we do know is that it has been there, in the dark, for a very long time. Longer, probably, than any of the scientists who discovered it have been alive. Growing silently, generation after generation, in water colder than a refrigerator, at pressures that would flatten a car, in a darkness so complete and so old that the concept of sunlight would be meaningless to every organism living in it. The ice broke on January 13th. The scientists arrived on January 25th. Sebastian went into the water, and the lights came on, and for the first time in the history of this planet, human eyes looked into that darkness and saw what was there. Section 3. What they found to really appreciate what the scientists aboard the R/V Falker 2 discovered beneath the George VI ice shelf in January 2025, you first need to understand what they were expecting to find. Or more precisely, what the scientific consensus at the time told them they should find. Before this expedition, the collective knowledge of what lives beneath Antarctica's floating ice shelves could be summarized with painful brevity. The ice shelves themselves cover more than 1.5 million square kilometers of the Antarctic continental shelf, an area larger than France, Germany, and Spain combined. They are among the largest and most persistent structures on the surface of the planet. And yet the scientific community's total direct observation of the seafloor beneath them, across all of human history and all of the expeditions that had ever tried, amounted to an area roughly the size of a tennis court. 8 boreholes, bored through hundreds of meters of ice at enormous expense and effort, had given scientists tiny windows into that world. 8 tennis court-sized glimpses into 1.5 million square kilometers of total darkness. What those glimpses had revealed before January 2025 was mostly what scientists expected: very little. Mobile scavengers, small fish, worms, krill, crustaceans, had been detected in a few locations. These animals can move. They can swim, crawl, or drift their way from the edges of the ice shelf into its interior, scavenging whatever organic material finds its way to them. Their presence was not surprising. The Southern Ocean is full of mobile life, and some of it was always going to find its way beneath the ice. What scientists had not expected to find, what the models and the theories and the prevailing logic of deep-sea ecology said could not be there, was stationary life. Filter feeders, animals that don't move, sponges, corals, anemones, hydroids, animals that survive by anchoring themselves to hard substrate and waiting for food to come to them, drawn from the water column by the biological machinery of their own bodies, flagella waving, pumping currents, filtering particles. These are animals that depend, fundamentally and completely, on a supply of organic material washing over them from some other place. They cannot go looking for food. Food has to come to them. And under the ice, where was that food supposed to come from? The ice above blocks sunlight. No sunlight means no photosynthesis. No photosynthesis means no phytoplankton bloom in the water column above. No bloom means no marine snow, the constant, essential rain of organic particles that sustains deep-sea ecosystems the world over. The supply chain was broken. The models said nothing should be there. The textbooks said nothing should be there. The first small crack in that certainty came in February 2021, when a team from the British Antarctic Survey published a paper that made marine biologists around the world sit up very suddenly in their chairs. The team had been drilling through the Filchneron ice shelf in the southern Weddell Sea, drilling with hot water through 900 meters of solid ice, in order to lower sediment coring equipment to the seafloor below. The plan was simple and methodical: pull up a core of mud, analyze the layers of sediment for their geological record of past climate and ice sheet behavior. Standard procedure. What happened instead was that the drill hit a boulder. The sediment corer came back empty. The frustrated geologist who had been living in a tent on the ice for 3 months eating freeze-dried food sent down a camera to see what the problem was. And what the camera showed, 260 kilometers from the nearest open ocean, in water at minus 2.2 degrees Celsius, with not a photon of sunlight anywhere in any direction, was a boulder covered in life. Not a hint of life, not a trace, covered. Sponges in at least 2 distinct forms: cylindrical growths, stalk protrusions, and what appeared to be hydroids and filamentous structures, and microbial mats spreading across the rock's surface like a living carpet. Creatures that should not, by any reasonable scientific framework, have been able to exist there. Hugh Griffiths, the British Antarctic Survey biogeographer who analyzed the footage, described the moment with the kind of eloquence that only comes from someone who genuinely cannot believe what they are looking at. Never in a million years would we have thought about looking for this kind of life, because we didn't think it would be there. But the 2021 discovery was limited in a very specific and frustrating way. It was accidental. The equipment that found the boulder was not designed to study biology. It was designed to core sediment. There was no ROV. There was no manipulator arm. There was no way to collect samples, no way to measure the animals, no way to sequence their DNA or determine what they were eating or how long they had been there. The scientists could only look at the camera footage from above and wonder. The discovery had cracked the door open on a world that no one had known existed, but it hadn't given them the tools to walk through it. What January 2025 gave the world, what the calving of A84 and the arrival of the Falcor 2 gave science, was finally, for the first time, those tools. And not just the tools, the opportunity, the sub-ice shelf environment, normally inaccessible to anyone or anything that needed to enter from the side because of the sheer extent and solidity of the ice, was now open from above. The ice roof had been removed. For the first time in the history of scientific exploration, an ROV could dive straight down into an ecosystem that had previously been as unreachable as the surface of another planet. Sebastian went in at the end of January 2025, and what it found was not a cracked door. It was a world in full bloom. The first thing the monitors in the control room showed, as Sebastian's lights swept across the seafloor at around 230 meters depth near the margin of where A84 had been, was a sponge. Not a small one, not a sparse, withered survivor clinging to the edge of existence in a hostile environment. A massive one, pale, elaborately structured, its filtration apparatus reaching upward in the water with the confident architecture of an organism that has been growing in this exact spot for a very, very long time. And sitting on top of it, clearly visible in Sebastian's lights, was a crab. The first thing we saw was a huge sponge with a crab on it, said Dr. Patricia Eskeet afterward. That was already quite surprising because one question we had was, will there be any life at all? That question, will there be any life at all, is the starting point of the story of everything that follows. The scientists aboard the Falcor, too, were not naive. They knew that the 2021 Filchneron discovery had suggested that life could persist in sub-ice shelf environments. But suggested is not the same as confirmed, and confirmed in one small boulder-sized sample is not the same as the full, complex, three-dimensional biological landscape that Sebastian's cameras began to reveal over 8 consecutive days of diving. The question hanging over every dive was not just whether there was life, but how much of it, how diverse, how old, and how deep it went in every sense of the word. The answer, delivered frame by frame as Sebastian's lights swept across hundreds of meters of seafloor and pushed deeper and deeper into the canyon systems that carved through the shelf slope, turned out to be extraordinary, staggering, unlike anything the scientific community had been prepared to imagine in that particular location. Let's go through what they found, one organism at a time, and try to give each one the space it deserves. Because each species that appeared on Sebastian's cameras that week was not just a data point. Each one was a story. Each one was a window into a world that had been sealed from human observation for longer than anyone alive has been alive. The sponges were, in many ways, the keystone of the entire discovery. Not because they are the most dramatic animals in the ecosystem. That competition has other contenders, but because of what they prove. Sponges are the oldest multicellular animals on Earth. Their lineage stretches back to somewhere between 600 and 700 million years ago, possibly longer. They appear in the fossil record at a point in time when the most complex life on the planet was still figuring out the concept of having distinct cells. They are extraordinarily simple in their organization. They have no nervous system, no muscles, no mouth, technically. Their entire body plan is essentially a highly sophisticated pumping and filtering apparatus. Water is drawn in through small pores called ostia, passed through an internal structure of water-filled canals, filtered of organic particles by specialized cells called choanocytes that capture and consume bacteria and fragments of organic material, and expelled through a larger opening called the osculum. That's it. That's the whole organism. Pump in water, extract food, expel water, grow. Because sponges are so simple, and because they feed passively on whatever the water brings them rather than actively pursuing prey, they grow slowly. Extremely slowly, in cold water. Antarctic sponges in the deep Southern Ocean are documented to grow at rates of less than 2 centimeters per year under favorable conditions. In the sub-zero, nutrient-sparse environment beneath a floating ice shelf, growth rates are likely even slower. This is why the size of the sponges that Sue Bastian photographed is so significant. Some of them were large enough that a person could wrap their arms around them and not meet their own hands on the other side. Sponges of that size, in those conditions, growing at those rates, have been alive for decades. Some almost certainly for a century or more. They are not recent colonizers who moved in after the conditions changed. They were not established in the last few years as the ice thinned and the environment shifted in subtle ways. They were there, growing in the darkness under the ice, when people alive today were children, when their grandparents were alive, quite possibly when their great-grandparents were alive. Sponges can grow very slowly, sometimes less than 2 centimeters a year. Therefore, the size of the specimens observed suggests these communities have been active for decades, perhaps even hundreds of years. That's not a poetic flourish. That's a scientific inference based on the direct observation of body size in organisms with known growth rates. Those sponges are living time capsules, clocks that measure not hours and minutes, but decades, possibly centuries. And they have been keeping time in total darkness, under the ice, in water that hovers just above the freezing point of salt, without anyone on the surface knowing they were there. Beyond their age, the sponges were doing something ecologically important that goes beyond their own biology. In the deep sea, large structural organisms, the things that build the scaffolding that everything else lives on, are called foundation species or habitat formers. A large coral colony on a tropical reef is a habitat former. A giant kelp frond in a temperate ocean is a habitat former. In the deep Antarctic subice environment, the sponges were playing that role. Their bodies, their structures, their skeletal frameworks, in many sponge species, composed of silica or calcium carbonate spicules arranged in intricate geometries, provided surfaces for other organisms to live on, hide in, feed from, and shelter within. The crab that appeared in that first moment of contact with the seafloor was not sitting on the sponge by accident. It was there because the sponge was its habitat. Take away the sponge, and the crab has nowhere to be. The sponge was not just an organism. It was a city. The corals and anemones scattered among the sponges, and particularly visible in deeper sections of the dives, were cold water corals. These are a different beast from the reef-building corals most people picture when they hear the word, the warm, shallow, brightly colored structures of tropical oceans. Cold water corals are solitary or colonial animals that live in the deep, cold, dark parts of the ocean all over the world, from the fjords of Norway to the seamounts of the deep Atlantic, and now, confirmed beyond any doubt, beneath the ice shelves of Antarctica. They are filter feeders, like the sponges, drawing organic particles from the water column. They can form dense aggregations, building up framework structures over decades and centuries. The cold water corals Sue Bastian observed in this environment were particularly striking because of their structural complexity. Cold water coral colonies don't grow by accident. Their branching forms, their accumulated calcified skeletons, their arrangement in the water column to maximize exposure to the passing flow of nutrients, are the product of sustained, incremental growth over long periods. You don't get a complex coral community in a year. You barely get one in a decade. The ones Sue Bastian documented spoke of a history measured in spans of time that make human planning horizons look embarrassingly short. Alongside and among the corals were sea anemones, relatives of corals and jellyfish, with those characteristic rings of flowing tentacles surrounding a central mouth. Anemones are both more mobile and more ephemeral than corals. Some species can creep slowly across hard substrate, and many are solitary rather than colonial. But what Sue Bastian's cameras documented was a profusion of them, clusters around the base of rocky outcroppings, individuals deployed at the rims of crevices, the feeding tentacles open and extended in the current, capturing whatever the water brought to them. Their presence, like the sponges and corals, was deeply counterintuitive in a place sealed from sunlight. They should not be there. They require food. They require a supply of organic particles flowing past them continuously enough to justify the metabolic expenditure of remaining alive. And they were there. The brittle stars and hydroid Sue Bastian's cameras also documented brittle stars, close relatives of sea stars, starfish, but with long, flexible, snaking arms that radiate from a small central disk, giving them a somewhat alien appearance when they move. Brittle stars are among the most abundant invertebrates in the deep Southern Ocean. They are scavengers and predators, using their arms to capture food particles, small animals, and carrion. One particularly striking image from the expedition showed a cluster of brittle stars climbing on a sponge at a depth of 215 meters, a perfectly composed tableau of the ecological relationship between foundation species and the organisms that depend on them. Drifting alone in the water column at around 380 meters depth was a solitary hydroid. Hydroids are among the most ancient and structurally interesting of the cnidarians, the same group that includes jellyfish, corals, and anemones. A solitary hydroid is a single polyp, not a colony. It exists alone in the water, anchored by nothing but its own buoyancy and the currents, extending its tentacles into the surrounding darkness in search of prey. Hydroids are related to corals and jellyfish, but form no colonies. The one Sebastian captured drifting in that soft underwater current, about as far from sunlight as it is possible to be and still be in an ocean, looked at once utterly alien and somehow profoundly, quietly alive. The krill at one moment during the dives. The footage showed something that struck viewers watching the live stream as almost comical in its everydayness. Swarms of krill clustering around Sebastian's lights. Krill, Euphausia superba, the Antarctic krill specifically, are among the most ecologically important organisms on Earth. They are small crustaceans, roughly 6 centimeters long, that form the foundation of the Southern Ocean food web, occurring in densities so immense that swarms can be detected from space by satellite ocean color sensors. Whales eat them, penguins eat them, seals eat them, squid eat them. Everything in the Antarctic ecosystem, it sometimes seems, either eats krill or eats something that eats krill. Their collective biomass is staggering. Estimates suggest that Antarctic krill represent the most abundant animal species on the planet by weight. But krill are not sedentary animals. They undergo daily vertical migrations, rising toward the surface to feed at night, sinking to depth during the day. They are driven by light. They are attracted to it, which is why they swarmed Sebastian's powerful lights. Krill descend under sea ice routinely. They scrape ice algae from the underside of sea ice as a food source. Their presence beneath the George VI ice shelf, while notable, was perhaps the least surprising element of the entire discovery. What was striking was seeing them there, in that particular darkness, in that particular place, clustering around the lights of a machine that had no business being there, performing the same phototactic behavior they would perform anywhere on Earth that a light source suddenly appeared in deep water. Their presence also, however, raised an important ecological point. If krill are present beneath the ice shelf, and they were clearly present, in visible numbers, then any predator that eats krill has a food source. The ecosystem is not as nutrient isolated as the models assumed. Krill can migrate under the ice from open water and bring with them a cargo of organic energy, in the form of their own bodies, that they scraped off the underside of sea ice or extracted from the productive surface ocean before diving. The sea spiders, but nothing prepared the science team, or the thousands of viewers watching the live stream on their phones and laptops on the other side of the world, for the sea spiders. Sea spiders, formerly known as pycnogonids, occupy one of the more surreal niches in the taxonomy of the animal kingdom. They are marine arthropods of the class Pycnogonida, not true spiders, nor even arachnids, despite the name and their superficial resemblance to the spiders most people know from land. There are over 1,300 known species distributed across every ocean on Earth, from intertidal tide pools to abyssal trenches, and most of them are small, cryptic little creatures hiding in coral rubble or algae, easily missed even by experienced marine biologists. The Antarctic variety is different. Most species of sea spider are smaller than a fingernail, but some Antarctic species have leg spans, tip of one leg to the tip of the opposite leg, of more than a foot. A foot. That is not a footnote to be skimmed past. That is a sea spider with legs that stretch farther than your hand can reach, with a body as big as a small apple, moving across the seafloor with a deliberate, slow-motion, alien grace that looks, in Sebastian's footage, exactly like what it is. One of the most physically improbable organisms on the planet. This phenomenon, Antarctic marine invertebrates growing to sizes far beyond what their temperate water relatives achieve, is called polar gigantism, and it is one of the most captivating and still somewhat mysterious observations in Antarctic biology. Polar gigantism is thought to have arisen from a combination of cold-driven low metabolic rates and high oxygen availability in the polar oceans, a framework researchers call the oxygen temperature hypothesis. The Southern Ocean, kept extremely cold by the circumpolar current and by Antarctica's proximity to the pole, is able to hold far more dissolved oxygen than warm tropical water. And cold temperatures slow down the metabolisms of ectothermic, cold-blooded animals, reducing the rate at which their tissues consume that oxygen. Put these two factors together and you create conditions where organisms whose body size is normally constrained by the difficulty of getting enough oxygen to large, metabolically active tissues can afford to grow large, because there's more oxygen available and they need less of it per unit of body volume. Sea spiders make this dynamic particularly vivid because they breathe through their cuticle, the hard outer surface of their exoskeleton, rather than through gills. There are no lungs, no gills, no specialized respiratory organ. Oxygen simply diffuses across the cuticle surface directly into the body. This system, called cutaneous respiration, works fine when an animal is small because the ratio of surface area to volume is high. There is enough cuticle to absorb enough oxygen for the relatively small interior mass. But as animals get larger, volume increases faster than surface area. A sphere's volume grows with the cube of its radius. Its surface area grows only with the square. Eventually, if you scale an animal up too far, the surface area can no longer supply oxygen to the volume, and the animal suffocates by slow degrees. Cold Antarctic water, with its extraordinary oxygen richness, relaxes this constraint. The cuticle of a giant Antarctic sea spider can supply oxygen to a body mass that would be lethally oxygen-starved in warm water. And the cold temperature means that body mass is using oxygen more slowly anyway. The result: leg spans over a foot, bodies the size of small mammals, moving through the water of the Bellingshausen Sea in the lights of Sebastian with a slow, inevitable, deeply strange deliberateness. To see one on the seafloor beneath the George VI ice shelf is to encounter the living proof that the universe of life on this planet is far stranger, far more varied, far more astonishing than the version of it most people carry around in their heads. See spiders of that size, in that darkness, at those depths. They are not supposed to exist in the imagination of someone who learned biology from textbooks with pictures of deer and maple trees. And yet there they are. There they were. The ice fish that appeared in Sebastian's footage are, by any measure, one of the most extraordinary fish on the planet. And they deserve considerably more attention than they usually receive, because their story is one of the great evolutionary dramas of the animal kingdom. Antarctica's crocodile icefish, members of the family Channicthiidae, are the only known vertebrates to lack hemoglobin in their blood as adults. Let that statement land for a moment. In the roughly 65,000 known species of vertebrate animals on Earth, fish, amphibians, reptiles, birds, mammals, every single one of them uses hemoglobin to transport oxygen through their blood. That red protein, built around an iron atom at its center, is essentially the invention that made complex vertebrate life possible. It allowed early fish to grow large, to become active, to develop the muscular, metabolically intense bodies that eventually gave rise to everything from sharks to elephants to human beings. Hemoglobin is not a detail of vertebrate biology. It is the foundation of it. The icefish discarded it. More recently, some biologists have proposed that the loss of hemoglobin was not a beneficial adaptation, but rather a genetic accident with unfortunate consequences. Since icefish blood can only transport 10% as much oxygen as typical fish blood, Ice fishes were forced to dramatically alter their bodies in order to survive. The blood of an ice fish is not red. It is colorless, not a drop of red anywhere. Their gills, which in normal fish are stained the dark, vivid red of oxygenated blood flowing close to the surface, are soft and white, like vanilla yogurt. And yet they are alive. Not just alive, thriving. Ice fish are among the dominant predators of the coastal Southern Ocean. They are active swimmers, with large heads and toothy jaws. They hunt. They reproduce. They are, by any functional definition, highly successful fish. They manage this impossible trick through a collection of compensatory adaptations so extensive and so comprehensive that they amount to a complete reinvention of vertebrate circulatory physiology. To compensate for the absence of hemoglobin, ice fish have adapted larger blood vessels, including capillaries and low viscosity, red blood cell-free blood to enable very high flow rates at low pressures. They also have greater blood volumes, 4 times those of other fish, and larger hearts with greater cardiac outputs, 5 times greater than comparable fish. Their hearts are enormous relative to their body size. Their capillaries are wide enough to let blood flow fast without the red cells that normally occupy a significant fraction of blood volume and create resistance. Their blood, stripped of red cells and the hemoglobin they carry, is essentially a thin, transparent liquid, easy to pump quickly, easy to circulate at high volume. And that high volume, fast circulating liquid draws oxygen not from hemoglobin, but directly from dissolution in the plasma itself, exploiting the fact that cold Antarctic water is so oxygen-rich that even blood without hemoglobin can dissolve enough of it to keep tissues alive. They also carry antifreeze glycoproteins in their blood, special proteins that prevent ice crystals from forming inside their cells even when the surrounding water is below the freezing point of normal biological fluids. Without these antifreeze proteins, the ice fish would literally freeze solid. They are protected by a biochemical innovation that Antarctic fish evolved somewhere between 6 and 20 million years ago as the Southern Ocean cooled, and which now makes them uniquely capable of surviving in waters that would kill most fish outright. When Sebastian's lights found ice fish resting on or near the sediment of the exposed George VI seafloor, it was seeing animals that represent perhaps the most dramatic evolutionary departure from the vertebrate blueprint that biology has documented. They are pale, long-bodied, slightly translucent in their fins and skin, with those characteristic large heads and eyes adapted for seeing in near darkness. They look, when you see them in footage, like something that was designed to inhabit exactly this place, this cold, dark, pressurized world beneath the ice, rather than accidentally ending up here through the chance of evolutionary drift. The octopus at 1,150 meters deeper in the expedition dives, as Sebastian descended into the gully systems that cut through the shelf slope like geological wounds, steep-sided trenches carved by ancient currents, and the slow movement of sediment. The seafloor changed character. It became more rugged, more complex, with rocky outcroppings interspersed among muddy sediment and biological gardens of corals and sponges growing on any hard surface available. And at 1,150 meters, nearly 3/4 of a mile below the surface of the Bellingshausen Sea, the cameras found something that became, in the days after the expedition's findings were made public, perhaps the single most striking image of the entire endeavor. An octopus, just sitting there. Octopuses are among the most intelligent, most curious, most behaviorally sophisticated invertebrates in the ocean. Their reputation for intelligence is well-deserved. They have large brains relative to their body size, sophisticated problem-solving abilities, excellent vision, despite being colorblind, and an uncanny capacity for behavioral flexibility that has been demonstrated in dozens of research studies. They can open jars. They recognize individual humans. They play. They dream, apparently. They have been described by some biologists as the closest thing on Earth to a fundamentally alien form of intelligence, cognitive sophistication that evolved entirely independently from the vertebrate lineage, in a body plan so different from our own that the experience of being an octopus must be almost incomprehensible from a human perspective. The octopus Sue Bastian photographed at 1,150 meters was not a small one. It sat on the seafloor with the fluid, settled posture of a large cephalopod at rest, arms deployed in a loose radial arrangement, body relaxed, mantle expanded. It appeared completely unperturbed by the arrival of Sue Bastian's lights. This is not unusual for deep-sea octopuses. They do not have the same instinctive avoidance of light that surface-dwelling animals develop when light means predators or exposure. In the permanent dark of the deep sea, light is a novel and potentially interesting event rather than a threat. What was that octopus doing there? It was, in all probability, doing exactly what octopuses do in every ocean at every depth they inhabit: hunting. The deep Antarctic seafloor beneath the ice shelf, with its sponge gardens and coral communities and anemone clusters, is also a place with small invertebrates, isopods, worms, small crustaceans, brittle stars, all of which are potential prey for a large, mobile, intelligent predator. The presence of an octopus at that depth is not just a remarkable sighting in itself. It is evidence of a food web complex enough to support a top invertebrate predator. You don't get an octopus somewhere unless there is enough prey to sustain one. The octopus is, in a sense, a certificate of ecological abundance. The squid consuming a fish at nearly 950 meters depth, Sue Bastian's cameras captured something even more predatorily dramatic. A squid, in the act of consuming a fish. The squid had seized the fish. The image shows it clearly, the fish gripped in the squid's arms, partially consumed, and it was not remotely bothered by the arrival of a 3-ton illuminated robot. This is how squid are. They are efficient, unsentimental predators, and efficiency does not have time for distraction. Squid are among the most ecologically important predators in the deep sea globally. They are fast, intelligent, by invertebrate standards, and equipped with powerful beaks capable of tearing through tissue, and with arms lined with suckers that can grip, hold, and manipulate prey with remarkable precision. They are also prey themselves for virtually every large vertebrate predator in the ocean: sperm whales, elephant seals, many seabirds. They sit in the middle of the marine food web, eating downward and being eaten upward, and their presence in large numbers beneath the George VI ice shelf is yet another signal that the ecosystem there is not a thin, marginal, barely surviving collection of organisms clinging to the edge of survivable conditions. It is a functioning, layered, predator and prey food web. It is alive in the most complete sense of the word. The giant phantom jellyfish, and then there was the thing that stopped the science team in the control room. Drifting through the water column in the area where the shelf break and slope are cut by the underwater gullies, not on the seafloor, but in the open water above it, at a depth that placed it squarely in the deep mesopelagic zone, was a giant phantom jellyfish, Stygiomedusa gigantea. With only around 118 sightings in 110 years, it is a jellyfish that is rarely seen, but believed to be widespread throughout the world, with the exception of the Arctic Ocean. The Monterey Bay Aquarium Research Institute's remotely operated vehicles have only sighted the jelly 27 times in 27 years. Think about that number for a moment. The Monterey Bay Aquarium Research Institute, one of the most prolific deep-sea ROV operation programs in the world, with thousands of log dives across decades of work, has encountered the giant phantom jellyfish 27 times. That's roughly once a year. Once in an entire year of deep-sea diving. The SROV encountered it during its very first expedition ever to this stretch of seafloor, on the first occasion any human being had ever set eyes on that particular piece of ocean. The bell of this deep-sea denizen is more than 1 meter across and trails 4 ribbon-like oral arms that can grow to more than 10 meters in length. The arms are not tentacles in the conventional stinging sense. The giant phantom jelly does not sting its prey. Instead, it wraps those 4 vast, velvety arms around its food, plankton, small fish, whatever drifts into range, and guides it upward toward its mouth. The arms move like silk scarves in a slow wind, trailing behind the pulsing bell with a mesmerizing, ghostly grace. When visible light is present, giant phantom jellies emit a slight orange-red light via bioluminescence, meaning they produce light through natural chemical reactions. In the deep ocean, where red wavelengths of light cannot penetrate from the surface, an animal that glows red is, counterintuitively, invisible. Red bioluminescence in the deep sea is a camouflage strategy, a way of glowing without being seen, of carrying a light source without advertising your presence to predators. The giant phantom jelly is, in that dim red glow, both beautiful and sinister. Giant phantom jellies are viviparous, meaning they give birth to live young. The young develop inside the mother before detaching from inside the hood and swimming out of their mother's mouth, which is a sentence that is difficult to process on first reading. Live young, swimming out of the mother's mouth. The biology of Stygiomedusa gigantea is, in almost every respect, a catalog of the unexpected. Evidence has been collected to support the first ever documented symbiotic relationship for an ophidiiform fish, Thalassobathia pelagica. Scientists have observed that the large umbrella-shaped bell of Stygiomedusa gigantea provides food and shelter for T. pelagica, while the fish aids the giant phantom jelly by removing parasites. The jellyfish is not just a predator. It is also a shelter provider, a mobile habitat in the open water, a relationship as intricate and mutually beneficial as any that exists in the sunlit shallow reefs of the tropics. It just happens to take place in pitch darkness a kilometer below the surface of the Bellingshausen Sea. For the scientists aboard the Falker, too, seeing the giant phantom jelly drift through the frame on their monitors was the moment the expedition transcended the merely extraordinary and entered the genuinely mythological. There it was, one of the rarest animals in the ocean, in a place that had been sealed under ice until 12 days before their arrival, glowing its faint orange-red glow in water as cold as the dark side of the moon. The possible new species throughout the 8 days of Sebastian's dives. The science team collected biological samples using the ROV's manipulator arms, carefully, methodically, capturing individual organisms and placing them in sample containers for transport back to the ship's laboratories. The physical process of collecting a specimen from the deep sea is painstaking. The manipulator arm must maneuver with enough precision to grasp a delicate organism without crushing it, at depths where the video feed is the only reference the operator has for position and contact. Among the samples collected, the team identified what appeared to be a new species of isopod, a crustacean in the group that also includes the common pill bug or roly-poly, though the deep-sea Antarctic isopods are larger, more complex, and considerably more alien in appearance. Isopods are enormously successful animals in the deep sea. They are scavengers, predators, parasites, and detritivores, and they occur in Antarctic waters in extraordinary diversity. The potential new species was photographed in a sample jar on deck, segmented, pale, chitinous, with the characteristic armored body plan of isopods rendered in an Antarctic form that science had, apparently, not previously named. Since the area became accessible, at least 6 new species have been identified in the region. That number, 6, confirmed at the time of publication, is almost certainly not the final count. The samples collected from those 8 dives will take years to fully analyze. Morphological description, genetic sequencing, comparison against museum specimens, and existing taxonomic literature. The slow, painstaking process by which marine taxonomy actually works, the process by which an organism moves from unusual specimen collected in this jar to formally described, named, and entered into the permanent scientific record of life on Earth. Some of the organisms photographed in Sebastian's footage, visible on the monitors but never directly sampled, may represent species known to science but previously unrecorded in this region. Others may represent entirely new lineages. The genetic work will tell. It will take years to know, and when it does, the number 6 will almost certainly be larger. What the ecosystem means as a whole step back from the individual organisms, from the sponges and the phantom jelly and the white-blooded fish and the octopus, and look at what they mean together. What they reveal as a collection, as a community, as an ecological structure spread across hundreds of meters of seafloor depth and 510 square kilometers of previously inaccessible ocean. What they reveal is an ecosystem, not a collection of isolated organisms that happen to share a space. An ecosystem in the full ecological sense, a web of relationships between organisms and their environment, involving energy flow, nutrient cycling, predation, competition, mutualism, symbiosis, succession, and all of the other processes that make a biological community something more complex than the sum of its parts. Sponges that create habitat for crabs and brittle stars. Corals that provide shelter and attachment surfaces for anemones and worms. Krill that provide food for ice fish and squid. Squid that provide food for octopuses. Octopuses that, well, octopuses presumably prey on whatever they can catch and avoid being prey themselves, which in the deep Southern Ocean is a challenge that involves outthinking rather than outrunning potential predators. And over all of it, drifting through the water column, the great ghost of the giant phantom jelly with its 10-meter arms trailing in the dark. Laura Samoli, a physical oceanographer at the University of Cambridge who led the oceanographic effort on the expedition, noted, there were many species occupying a variety of ecological niches and with a variety of functional traits. Ecological niches, functional traits. This is the language of an ecologist describing a complex, differentiated biological community, not a marginal one. A community with structure, with layers, with the kind of internal complexity that takes time and stability and a sustained supply of energy to build. Which brings everything back to the question that hung over the entire expedition, the question that the scientists aboard the Falcor, too, could not answer before leaving. The question that the gliders and the sediment cores and the water samples they collected will spend years helping to untangle. How? How is this ecosystem here? How has it been sustained, in the dark, under the ice, cut off from the surface productivity that deep-sea ecology textbooks say it needs? How have the sponges grown to their current size? How are the corals building their structures? What are the ice fish eating? What is feeding the krill? Where does the organic material come from that cycles through this community, from bacterium to sponge to crab to ice fish to octopus and back again to bacterium? The answer to that question, which is the answer to the mystery at the heart of this entire story, takes us somewhere even more interesting. And it is where we turn next. Section 4. The mystery. There is a principle in science, not a law, not a theorem, but a guiding intuition that researchers tend to operate with, especially in ecology, that if you want to understand an ecosystem, you begin by understanding its energy. Everything else follows from that. Population sizes, species diversity, the structure of the food web, the complexity of the relationships between organisms, all of it is downstream, in a very literal sense, of the question, where does the energy come from? For almost all of the visible life on Earth, the answer to that question is the sun. The sun is the engine. It has been the engine for as long as complex life has existed on this planet. Even in situations where the connection is not immediately obvious. Even when you are eating a piece of beef from a cow that ate grain from a plant that grew in a field that was tended by machines powered by petroleum extracted from rock, if you trace the chain of energy transfer all the way back to its source, you arrive, eventually, at sunlight. The petroleum is fossilized sunlight. The grain was made from sunlight. The cow converted that sunlight. You are, in a very real sense, made of sunlight that fell on the earth millions or hundreds of millions of years ago and was captured, converted, and passed from body to body in an unbroken chain down to you. Even deep-sea ecosystems, in the cold, dark places where the sun's light has never penetrated, are connected to this chain. The marine snow that rains down through the water column, those tiny particles of organic material, dead algal cells and fecal pellets and bacterial biomass and the disaggregated fragments of organisms that died in the sunlit surface ocean, is the same chain, extended. Surface productivity in the sunlit zone, driven by photosynthesis, produces biomass. That biomass gets eaten and eaten again, and eventually its remnants fall toward the seafloor as marine snow. The deep sea lives on the leftovers of the surface. It has always done so. The energy that powers the worm burrowing through the abyssal sediment of the Pacific came, ultimately, from sunlight that hit the surface of the ocean sometime in the past few months or years. This is the picture that marine biologists and oceanographers had assembled by the early 20th century, and it held up well for decades. Deep-sea ecosystems were real, and they were studied, and they were understood to be dependent on the surface, poorer, simpler, and more thinly populated than shallow water ecosystems because the supply chain was long and inefficient. Energy is lost at every step in a food chain, which is why ecosystems get less rich and less diverse the further you get from the primary source. The deep sea was the end of the chain, the bottom of the barrel, the place where the last dregs of solar energy finally got converted into the bodies of pale, slow-moving organisms in the dark. And then, in 1977, a research team in the submersible Alvin dove to a depth of 2,500 meters along the Galapagos Rift in the eastern Pacific, and everything changed. The discovery of hydrothermal vent ecosystems in 1977 is one of the most important moments in the history of biology, and it does not receive anywhere near the attention it deserves in popular culture. Biology, who had come to study the thermal gradient along a mid-ocean spreading ridge, found something that should, by every principle of ecology then established, have been impossible: a dense, thriving, richly diverse community of organisms clustered around seafloor vents that pumped superheated, chemically enriched water into the deep ocean. Giant tube worms, some as long as 2 meters. Dense beds of clams the size of dinner plates. Mats of white bacteria covering every surface. Shrimp, crabs, fish. Instead of sunlight being the primary form of energy, chemical energy was being used in a process called chemosynthesis. Bacteria at the base of the vent food web were harnessing the energy locked in hydrogen sulfide, a toxic gas, the chemical responsible for the smell of rotten eggs, bubbling up from the hot vents, and using that chemical energy to fix carbon, building organic molecules from scratch without any input from the sun whatsoever. Life at vents proved that chemosynthesis could support thriving communities completely independent of sunlight. This revelation transformed the search for life elsewhere in the solar system. This was genuinely revolutionary. Not the revolutionary that science press releases used to describe incremental advances, but actually, structurally, paradigm shifting. Before 1977, the operating assumption of biology was that all complex life was, at some remove, solar-powered. After 1977, that assumption was false. Life did not require the sun. Life required energy, and energy could come from sources that had nothing to do with the star at the center of our solar system. The universe of possibilities for where life could exist, and on what terms, expanded enormously in the decade or so after the Galapagos Rift discovery. But the vent ecosystems had a specific, identifiable energy source: chemical energy from the Earth's own internal heat, expressed as superheated, mineral-rich water through cracks in the seafloor. They were not simply dark, they were dark and chemically powered. The ecosystem beneath the George VI ice shelf is different. There are no identified hydrothermal vents in the area where Sebastian dove. The seafloor geology of the Bellingshausen Sea's continental shelf is not characterized by the kind of active volcanism and seafloor spreading that drives hydrothermal activity. The organisms Sebastian found, sponges, corals, sea spiders, ice fish, are filter feeders and predators adapted to ecosystems that run on organic energy. The carbon-rich bodies of organisms, the dissolved organic matter produced by biological activity. They are not the sulfur-processing tube worms and specialized chemosynthetic clams of the vent ecosystems. They are conventional, if extraordinary, deep-sea metazoans, the kind of animals that live in deep-sea ecosystems all over the world, but always, everywhere else, in deep-sea ecosystems that receive marine snow from above. Here, there is no marine snow from above. There has been no marine snow from above for an unknown, but certainly long period of time. The ice sealed the sky. So the question is not, could chemosynthesis explain this ecosystem? The honest answer to that question is, possibly in part, at the microbial level, but not for the filter-feeding sponges and the corals and the ice fish and the octopuses. Those animals require organic food delivered through the water. They need a rain of particles or a current bearing dissolved and particulate organic matter or prey animals moving through their space. They need the food web's currency, organic carbon, to come from somewhere. And the mystery is, where? To understand the candidate explanations that the scientists on the Falcor, too, considered, and that oceanographers and biologists studying the data from the expedition have been wrestling with since, you need to understand something about the particular oceanography of the region beneath an Antarctic ice shelf, because it is not simply a static, sealed environment. It is a place where physics is doing interesting and still incompletely understood things. When an ice shelf sits on the ocean, floating, grounded at its landward edge, drifting free at its seaward face, it is not inert. It is melting at its base, at a rate that varies depending on the temperature of the water directly beneath it, the geometry of the subshelf cavity, and the circulation patterns within that cavity. The underside of the George VI ice shelf is in contact with ocean water, and that water is doing what water does when it contacts ice that is slightly above the melting point. It melts it, slowly, continually. The melt rate is not dramatic. We are not talking about ice cubes in a warm drink. We are talking about meters per year, at most, sustained over decades and centuries. But even a slow melt, sustained over long enough time, produces a significant and continuous flux of fresh water rising from the ice surface into the subshelf ocean cavity below. This freshwater meltwater is physically different from the salty seawater around it. It is less dense. Fresh water, at the same temperature, is lighter than salt water. So it rises. It forms a buoyant plume along the underside of the ice, flowing upward and then outward, mixing with the surrounding seawater as it goes. And as it rises and flows, it drives circulation. The physics of buoyancy-driven flow under an ice shelf is complex. It depends on the slope of the ice base, the temperature of the surrounding water, the salinity gradients within the cavity, but the fundamental dynamic is clear. Meltwater creates motion, and motion in the subshelf cavity is what connects the isolated world beneath the ice to the larger ocean outside. Dr. Laura Simoli, from Cambridge's Department of Applied Mathematics and Theoretical Physics, who led the oceanographic effort on the expedition, noted, "The fact that we found long-living species suggests that the lateral transport, which mostly consists of glacial meltwater from the ice shelf, could be the source of the nutrients to sustain the life we found." Lateral transport. That phrase is doing an enormous amount of work in the scientific conversation around this discovery, and it is worth spending some time with it. Lateral transport, in oceanographic terms, means the horizontal movement of material through the ocean, as opposed to vertical transport, which is marine snow falling from above. In most deep-sea ecosystems, vertical transport is the dominant mechanism by which organic matter reaches the seafloor. But beneath an ice shelf, where vertical supply is blocked, the possibility of lateral supply, organic material flowing in horizontally, along the seafloor or through the water column, from somewhere outside the ice shelf boundary, becomes the primary mechanism by which the food web could possibly be sustained. The gliders that the Falcor, 2, team deployed in the area before leaving, those autonomous underwater vehicles that move through the water column taking continuous measurements, returned preliminary data suggesting something important. The glider data showed high biological productivity and a strong meltwater flow from the George VI ice shelf. Biological productivity, measured in the oceanographic sense, concentrations of chlorophyll and other pigments that indicate active photosynthesis, concentrations of dissolved organic carbon, measurements of community respiration rates, was higher in this region than the sealed under ice environment would naively suggest. And it was correlated with the meltwater signal. Wherever the meltwater was flowing, productivity appeared to be elevated. This is a clue, but it is not yet a complete explanation. The fact that productivity correlates with meltwater flow could mean several things. It could mean that the meltwater itself is carrying nutrients from the melting ice. Iron, for instance, which is often the limiting nutrient for phytoplankton in the Southern Ocean, in which glacial meltwater is known to transport in elevated concentrations. Iron released from melting ice fertilizes the surface ocean in ways that have been documented extensively around Antarctic ice margins. But this fertilization happens at the surface, where there is sunlight to drive the photosynthesis that iron enables. Under the ice, there is no surface, no sunlight. The iron from the meltwater cannot drive photosynthesis if there is nowhere for photosynthesis to happen. Unless, and this is one of the more intriguing possibilities being considered, the meltwater is not merely delivering nutrients. It is delivering them somewhere, and the motion it creates is connecting the subshelf environment to a larger circulation system that extends beyond the ice shelf boundary to places where sunlight does reach the surface and where biological productivity is occurring. The key to this hypothesis lies in understanding how the Southern Ocean moves, and specifically how it moves around the Antarctic continental margin. The Southern Ocean is one of the most dynamically active bodies of water on Earth. It is the only ocean that circles the globe without interruption by any continental landmass, and this continuous circumpolar pathway, the path of the Antarctic Circumpolar Current, the strongest ocean current on the planet, drives a global-scale mixing and overturning of the world's oceans that is fundamental to Earth's climate. Antarctic bottom water, the cold, dense, oxygen-rich water mass produced when brine is rejected during sea ice formation on Antarctic continental shelves, sinks to the bottom in regions like the Weddell Sea and then moves north along the bottom into the Atlantic and east through the Southern Ocean. It is, in the language of oceanographers, the deepest and densest component of the global thermohaline circulation, the system sometimes called the global ocean conveyor belt, though that metaphor simplifies something far more complex and three-dimensional than any conveyor. As the densest water mass of the oceans, Antarctic bottom water occupies the depth range below 4,000 meters of all ocean basins that have a connection to the Southern Ocean at that level. This is the water that was cold and dense enough, when it was formed near the surface at the Antarctic margin, to sink all the way to the bottom of every major ocean basin on Earth. It is the water that ventilates the abyss. It carries oxygen from the surface, where it was last in contact with the atmosphere, down into depths that would otherwise stagnate. It also carries, dissolved in its volume, the signature of where it came from, the cold, nutrient-rich, biologically productive continental shelves of Antarctica, where sea ice formation and meltwater dynamics drive some of the most intense biological productivity in the global ocean. Now imagine this: the Antarctic coastal current, the west-flowing current that runs along the Antarctic continental margin, sweeps productive surface waters westward along the coast. As it does, it mixes organic material produced in the sunlit coastal polynyus, the ice-free patches of ocean along the Antarctic coast where biological productivity can be intense, where phytoplankton bloom in summer with extraordinary vigor, gets incorporated into the coastal current. Some fraction of this material, mixed and stirred by the turbulent coastal flow, eventually enters the subshelf cavities, not directly from above, but from the edges, carried horizontally under the ice shelf's seaward margin by the flow of the water itself. By studying current charts surrounding the Filchneron drill site, researchers concluded that there are abundant food sources between 390 and 930 miles away, and that a stream of organic material drifting amid these currents might be enough to feed these hungry creatures, even though it seems almost inconceivable when considering the vast distance. And the marine biologist Rich Mui captured the essential truth about Antarctic oceanography that makes this less implausible than it sounds. It's not that far-fetched when understanding how complex Antarctic waters are the engine that runs Earth's oceanic currents. The Southern Ocean is not a passive backdrop to the ecosystem mystery. It is an active participant. Its currents, its density-driven overturning, its extraordinary mixing dynamics. These are the mechanisms by which organic material could potentially travel hundreds of kilometers from a productive coastal polynya. beneath the floating boundary of an ice shelf and arrive at a seafloor 1,300 meters below the surface, carrying enough carbon and nitrogen and other biological currency to sustain a sponge that has been growing in that spot for a century. But how much material? And in what form? And at what rate? These are the quantitative questions that the expedition left unanswered, because answering them requires a different kind of sampling campaign than an 8-day ROV dive program. To understand the flux of organic material into a subshelf cavity, you need moored instruments that can measure current speed, direction, and particle concentration continuously over months or years. You need sediment traps, devices that catch and accumulate the particles sinking through the water column, deployed at multiple depths in the subshelf environment. You need stable isotope analysis of the organisms themselves, which can act as chemical fingerprints revealing where their food came from. A sponge whose body is built from organic matter that originated in a coastal polynya photosynthetically will have a slightly different ratio of carbon, 13 to carbon, 12 than one built from chemosynthetic organic matter, or from material derived from meltwater-carried dissolved organic carbon. The isotopes tell the story of the food web's origins in a language that no camera can capture. None of that was possible to do on the January 2025 expedition. The team had 12 days warning that the opportunity existed. They had a ship that was configured for seafloor mapping and biological sampling. They had Sebastian. They did extraordinary work with the tools they had. But the tools for resolving the energetic mystery, the instruments needed to quantify the flux, characterize the sources, and measure the rates, were not on the ship. They would require a dedicated follow-up expedition, planned in advance, equipped specifically for the purpose. The Antarctic is changing rapidly, Dr. Eskeet said after the expedition. And in order to understand what was going to happen, we really need to come back and keep studying and keep trying to learn and understand what was driving that ecosystem under the ice shelf. Come back. Keep studying. Keep trying to learn. These are not the words of a scientist who thinks the problem is nearly solved. They are the words of someone who has opened a door and seen how large the room on the other side of it is. There is a second candidate explanation for how the ecosystem sustains itself that has received less attention in the popular coverage of the A84 discovery, but deserves serious consideration, and that is the role of sea ice itself as a seasonal delivery mechanism. Antarctic sea ice, the seasonal ice that forms each winter on the surface of the Southern Ocean and retreats each summer, is not biologically inert. Its underside is colonized by ice algae, photosynthetic microorganisms that grow in the brine channels threading through sea ice, exploiting the thin light that penetrates from above even through several meters of frozen ocean. These ice algae are important organisms in the Southern Ocean food web. They are eaten by krill, which scrape the algae directly off the ice's underside. They bloom at the margins of sea ice in spring, when the ice begins to melt and their concentrated biomass is released into the water column in a pulse of biological material that fuels one of the most productive seasons in the Southern Ocean ecosystem. Now, the George VI ice shelf is bordered by the Bellingshausen Sea, which has significant seasonal sea ice formation and retreat. In winter, sea ice advances across the surface of the Bellingshausen Sea, potentially extending over or near the seaward margin of the ice shelf. In spring and summer, that sea ice retreats, and as it does, it releases its cargo of ice algal biomass into the water column. Some fraction of that material, carried by currents along the continental margin and potentially drawn into the subshelf cavity by the same buoyancy-driven circulation driven by basal melting of the ice shelf, could deliver a seasonal pulse of organic carbon to the ecosystem below. The krill that Sebastian observed clustering around its lights in the subshelf environment may be the clearest evidence for this mechanism. Krill perform vertical migrations, rising and sinking in the water column in response to light, and they are well documented to move under sea ice in search of ice algae. Their presence in the subshelf environment suggests they are accessing the space from its seaward opening, which means the seaward opening is accessible, which means organic material carried by currents and migrating organisms can enter from the open Bellingshausen Sea. The krill themselves are a vector. Their bodies carry the energy of ice algae and surface phytoplankton from outside the ice shelf into the darkness within, making that energy available to predators, ice fish, squid, octopuses that can hunt them. This does not fully explain the deep filter feeders, the sponges and corals and anemones at depths of 230 to 1,300 meters, whose feeding strategy depends on particles washing past them in the water flow rather than prey they can actively pursue. But it suggests a more complex and multi-pathway energy delivery system than a simple lateral diffusion model would predict. The ecosystem beneath the ice shelf may be sustained not by a single mechanism, but by several simultaneous ones, each contributing a portion of the energy budget. Lateral transport of dissolved organic carbon in the currents from distant productive areas. Seasonal pulses of ice algal biomass carried from the retreating sea ice margin. Mobile organisms like krill bridging the gap between the productive open ocean and the sealed subshelf cavity, and possibly even some local chemosynthetic contribution from microbial communities in the sediment at the deepest levels. A web of supply lines, all individually inadequate, that together add up to enough. One of the most striking pieces of evidence that the ecosystem had been receiving a sufficient and consistent energy supply for a very long time comes not from the biological samples themselves, but from what the biological community looked like as a whole. When ecologists describe an ecosystem as having distinct ecological niches and a variety of functional traits, they are describing something specific, not just a collection of organisms, but a community that has differentiated over time into the kind of structured, layered, role-specialized assemblage that only develops when conditions have been stable enough, for long enough, to allow the process of ecological succession to run most of its course. Dr. Eskeet noted that the team found several species filling discrete ecological niches, which suggested that the ice-covered ecosystem received a steady, high-level influx of nutrients and may have been there for a while. Basically, we found the same type of ecosystems that you can expect in that area of the Bellingshausen Sea, she said. But unlike the other areas the team studied, this ecosystem thrived in an area that's been permanently covered by ice for probably centuries. That statement is worth sitting with. The same type of ecosystem you would expect in the open Bellingshausen Sea. The same, not a diminished version, not a marginal, impoverished, barely hanging on version. The same richness, the same diversity, the same structural complexity, in an area that has been permanently covered by ice for probably centuries. This is either a coincidence that stretches credulity, or it is telling us something profound about the resilience of biological communities and the capacity of Antarctic oceanography to sustain life in places that our models told us it could not. The ecosystem did not appear to be struggling. It appeared to have adapted, completely and successfully, to the conditions of its sealed-off world, developing its own internal logic, its own food web structure, its own ecological niches, its own energy economy, in total isolation from the surface that most of us think of as the origin of all biological activity. There is a concept in ecology called the baseline, the state of an ecosystem before significant human disturbance. Baselines matter enormously in conservation biology because they tell you what you are working toward restoring, and because without knowing what an ecosystem looked like before humans started changing it, you have no reference point against which to measure loss. One of the tragic features of modern ecology is the phenomenon sometimes called shifting baselines. Each generation of scientists growing up with a diminished version of the world as their reference point for normal, not realizing how much had been lost before they started paying attention. The ecosystem beneath the George VI ice shelf is, in a very specific and important sense, the ultimate baseline. It has not been fished. It has not been exposed to agricultural runoff, microplastics, ship traffic, light pollution, noise pollution, or any of the dozens of other forms of human disturbance that have altered virtually every other marine ecosystem on Earth. It has been sitting in its sealed cavity, completely isolated, for centuries, doing exactly what it would have done without human civilization. Whatever species composition, whatever size structure, whatever predator-prey dynamics it has arrived at. These are, as close as we can get, the natural equilibrium state of a deep-sea Antarctic shelf ecosystem. And what happens now? A84 has drifted away. The ice is gone. The sealed cavity is no longer sealed. Sunlight will now, for the first time in centuries, filter through the surface of the Bellingshausen Sea above the George VI seafloor. Phytoplankton will begin to grow. Marine snow will begin to fall. The first photosynthetically produced organic material to ever reach this particular seafloor in living memory will start to drift down. Scientists hope to return to the site to investigate how that patch of seafloor changes now that its icy cover has drifted off. Nutrients trickling down from photosynthetic algae might now be available, but the ecosystem has already adapted to and thrived on a lower nutrient supply. As climate change melts Antarctic ice, this ecosystem could be a bellwether for changes across polar ecosystems. Will the ecosystem benefit from the increased food supply? Will the species that have adapted to low nutrient conditions over centuries be outcompeted by faster-growing organisms that colonize from the open ocean now that the ice barrier is gone? Will the warming of the water column, already occurring in the Bellingshausen Sea at rates faster than the global ocean average, stress organisms adapted to near-freezing temperatures? Will the sponges, those centuries-old living archives of Antarctic biological history, be overwhelmed and die as the conditions that formed them change faster than they can adapt? Nobody knows. The scientists would have to come back. They would have to watch. The mystery of how the sub-ice shelf ecosystem sustains itself is not just a puzzle about this particular patch of ocean floor. It is a question that has implications for our understanding of life across the entire 1.5 million square kilometers of seafloor that lies beneath Antarctic ice shelves. If the mechanisms at work beneath the George VI ice shelf, lateral transport, meltwater-driven circulation, seasonal inputs from sea ice, mobile organisms bridging the gap between light and dark, can sustain the kind of rich community Sebastian documented, then those same mechanisms are presumably operating at varying efficiencies beneath every ice shelf that covers Antarctic continental shelf seafloor, beneath the Ross Ice Shelf, beneath the Filchneron, beneath the Getz, the Dotson, the Crossen, the Pine Island, beneath all of them. If life can thrive at the 1,300-meter depths Sebastian documented beneath A84's former territory, with no direct solar energy supply, sustained entirely by the physics and chemistry of the ocean around it, how many other communities like this are there? How many sponge gardens, centuries old, are sitting right now in the permanent dark beneath ice shelves that no calving event has yet exposed? How many species are there, in the ecological inventory of life on Earth, that science has never encountered because the only way to encounter them would require a city-sized iceberg to break off and a research vessel to be close enough to get there in 12 days? The answer to those questions is unknown. It may remain unknown for a long time. Antarctica is enormous, remote, and expensive to study. Most of its continental shelf remains unsampled. Most of its subshelf ocean has never been touched by any scientific instrument. The full inventory of what lives beneath Antarctic ice is almost certainly not merely larger than what was known before January 2025. It is, the January 2025 discovery suggests, qualitatively different, richer, more complex, and more ancient than the prevailing models assumed. There is one more dimension to the mystery that is worth considering before moving on, and it is the most philosophically interesting of all. It concerns not what is eating what, or how organic material moves through the water column, but what it means, cognitively, conceptually, for how we understand life. That a community of organisms can thrive for centuries in conditions we would unhesitatingly describe as extreme, isolated, hostile, and impossible to sustain. We tend to define life's requirements by the requirements of the life we know best, our own, and the ecosystems we grew up with and studied first. Sunlight, warmth, moderate pressure, a clear sky overhead, and a reliable supply of nutrients drifting down from above. These are the conditions of the surface world, and they are the conditions that most biology textbooks were written around. They are also, in the full context of the planet, quite specific. quite unusual, quite dependent on the particular accident of being in the right place, near the surface, in the light, in the moderate temperature zone, that describes a small fraction of the total volume of the biosphere. The world beneath the George VI ice shelf is not in the light. It is not warm. Its pressure would flatten a car. Its temperature hovers within a degree of the freezing point of salt water. Its food supply is uncertain, indirect, and apparently adequate. And it has been producing, sustaining, and complexifying a community of living organisms for what is probably centuries without any of the conditions we default to thinking of as necessary. The sponge that Dr. Eskeet saw on Sebastian's first dive, the huge one with the crab perched on top, did not know it was in the wrong place. It did not know its conditions were supposed to be hostile. It just grew, millimeter by millimeter, year after year, in the dark, in the cold, in the sealed and silent space beneath the ice, eating whatever the currents brought it, building its silicate skeleton one microscopic layer at a time, living out its centuries with the total equanimity of an organism that has never been asked whether its environment is suitable and does not care what the answer would be. That is the mystery, really, not the specific mechanism of lateral transport versus meltwater chemistry versus seasonal krill delivery. Those are the mechanical details. The real mystery, the one that the discovery of A84's hidden world puts at the center of biology's agenda, is this. What are the actual limits of life? Not the limits we assume from the ecosystems we happen to study first, but the real limits, tested in the places we have never looked? We have, it turns out, barely begun to ask that question on Earth. And the answer, every time we manage to look somewhere new, seems to be, the limits are further than we thought. Section 5. The subglacial lakes, everything we have talked about so far. The calving of A84, the sprint of the R/V Falker, 2, to reach the newly exposed seafloor, the 8 days of dives in which Sebastian descended into a world of sponges and ice fish and giant phantom jellyfish. All of it took place in the ocean, in salt water, beneath a floating ice shelf, at depths ranging from a few hundred to 1,300 meters. The ice above those ecosystems was real and it was thick, but it was floating ice, a shelf, not a sheet. And the ocean beneath it, however dark and inaccessible, was connected, however indirectly, to the larger Southern Ocean outside. But Antarctica has another hidden world entirely, one that is not connected to the ocean in any direct way, one that has been sealed not for decades or centuries, but for thousands, tens of thousands, in some cases millions of years. One that is not beneath a floating ice shelf, but beneath the ice sheet itself, the enormous, continental-scale mass of glacial ice that sits on top of the Antarctic landmass and that, if it melted entirely, would raise global sea levels by approximately 60 meters. Beneath that ice sheet, in the darkness under kilometers of compressed glacial snow, hidden from sunlight and from the atmosphere and from every form of the surface world that most life on Earth depends on, there are lakes, hundreds of them. The existence of liquid water beneath the Antarctic ice sheet is, when you first encounter it, deeply counterintuitive. Antarctica is the coldest continent on Earth. It holds approximately 70% of all the fresh water on the planet in its frozen form. Its ice sheet in places is more than 4 kilometers thick, thick enough that the surface of the ice sits at an altitude of over 3,000 meters above sea level. The average annual temperature at the South Pole is around minus 49 degrees Celsius. The idea that anything beneath all of that could be liquid seems, at first pass, physically impossible. But physics does something interesting when you pile 4 kilometers of ice on top of something. Ice is heavy. A column of ice 4 kilometers tall exerts an immense pressure on whatever sits at its base. A pressure so great that it lowers the melting point of ice below the standard 0 degrees Celsius. This is the same phenomenon that allows ice skates to work. The pressure of the blade on the ice lowers the melting point just enough to create a thin film of liquid water that reduces friction. Scale that affect up to the weight of the Antarctic ice sheet pressing down over millions of square kilometers of bedrock, and you create conditions where water at the base of the ice sheet can exist in liquid form even at temperatures well below zero. That pressure-driven lowering of the freezing point is only part of the story. The Earth's interior is warm. Geothermal heat flows upward through the planet's crust everywhere on Earth, including beneath Antarctica, at a slow but continuous rate, typically about 50 to 60 milliwatts per square meter in the Antarctic interior, though it can be considerably higher in regions of geological activity. Over millions of years, that slow geothermal warmth, rising up through the bedrock and meeting the base of the overlying ice sheet, has melted ice from below. The meltwater pools in topographic depressions in the bedrock, valleys, basins, the low points between subglacial mountain ranges, and accumulates into lakes. Sealed above by kilometers of ice, sealed below by ancient rock, sealed on all sides by frozen walls and geologic time. The first hint that any of this was happening beneath Antarctica came in 1959 and 1964, when a Russian geographer named Andrei Kapitsa carried out seismic surveys of the ice sheet during two separate Soviet Antarctic expeditions. Kapitsa's primary goal was simply to measure the thickness of the ice sheet, and the technique, detonating small explosives and measuring the travel time of the resulting seismic waves through the ice to the bedrock below, was the standard method of doing so. But when Kapitsa analyzed his data from a region of East Antarctica near what would become Vostok Station, he found something strange. The signal from the base of the ice sheet was unusually flat, unusually smooth, unusually reflective. Not the irregular, rough return you would expect from ice meeting bedrock, but the kind of clean, mirror-like reflection that physicists associate with one specific kind of interface: ice meeting liquid water. Kapitsa filed his data and his interpretation away. The Soviet scientific system did not always facilitate rapid international publication of these kinds of findings. It was not until the 1990s, when British and Russian scientists jointly reanalyzed the accumulated radar data from decades of overflights above Vostok Station, that the full picture emerged. In 1996, the discovery was published in the journal Nature. Beneath Vostok Station, under approximately 4 kilometers of ice, there was a lake. Not a pond, not a seep, not a modest pool of meltwater. A lake 240 kilometers long, 50 kilometers wide, roughly the same surface area as Lake Ontario, the 16th largest lake in the world by surface area, sitting in complete darkness under 4 kilometers of ice. Its water estimated to have been sealed from the atmosphere for somewhere between 15 million and 25 million years. Lake Vostok, the largest subglacial lake on Earth. The discovery of Vostok transformed the way glaciologists thought about Antarctica's ice sheet, and it did so almost immediately. If a lake the size of Ontario could exist undetected under 4 kilometers of ice for the entirety of recorded human history, then what else was down there? The question was answered with extraordinary speed once researchers knew what they were looking for. Radio echo sounding, the technique of flying aircraft over the ice sheet while broadcasting radio waves downward and measuring their reflections, had been used to map ice thickness for decades. When researchers went back to those accumulated datasets and specifically looked for the flat, bright reflections that signaled liquid water at the base of the ice, they found them everywhere. Lakes under Dome Sea in East Antarctica, lakes under the ice streams of West Antarctica, lakes scattered across the continent like puddles after a rain, some large, most small, all of them invisible from the surface, and all of them liquid. The current inventory of Antarctic subglacial lakes exceeds 675 known bodies of water. Over 400 of them are considered sufficiently well characterized to be included in formal catalogs. The largest, as established, is Vostok, but the inventory includes lakes of every size down to features barely larger than a city block. Some are deep. Lake Vostok's southern basin may reach depths of 1,000 meters or more. Others are shallow, barely 10 or 15 meters from floor to the ice ceiling above, more like flooded caverns than open lakes. Some appear to be completely stable and isolated, their water sitting in the dark, essentially unchanged for geological epochs. Others are startlingly dynamic. They fill and drain on timescales of months to years, exchanging water with neighboring lakes through subglacial conduits and channels, sometimes releasing massive floods of meltwater that travel under the ice sheet to the ocean, lubricate the base of ice streams, and affect the speed at which glaciers move. Together, The subglacial lakes of Antarctica contain a significant fraction of all the liquid fresh water on Earth. The total volume locked in their darkness is difficult to estimate precisely, but Lake Vostok alone is thought to hold somewhere between 5,000 and 65,000 cubic kilometers of water, enough to fill the Grand Canyon and have a substantial amount left over, by at least 25%. And in all of that water, in all of those lakes, separated from sunlight by kilometers of glacial ice, from the atmosphere by the same barrier, from surface life by millions of years of isolation in some cases. What lives there? Before anyone could attempt to answer that question directly, they had to confront a challenge so formidable that it occupied the scientific community for the better part of 2 decades before any direct sampling was attempted. The challenge was this: how do you get into a subglacial lake without contaminating it? The lakes are pristine. They have been sealed for so long, isolated so completely, that they constitute some of the last truly uncontaminated natural environments on Earth. Any life that exists in them has evolved in total isolation from surface organisms, developing biochemistry and genetics shaped by millions of years of adaptation to conditions that differ radically from anything at the surface. If you drill a hole down to a subglacial lake and in the process carry even a single surface microorganism, on the drill, in the drilling fluid, on the equipment that enters the borehole, you risk introducing a foreign organism into an ecosystem that has no defenses against it. The contamination of a subglacial lake would not just be a scientific mistake, it would potentially be an irreversible ecological one. The introduction of an invasive organism into an isolated biome that has no previous exposure to it and no evolved response. The protocols developed to prevent this contamination are extraordinary in their rigor. Equipment is subjected to UV sterilization. Drilling water, the hot water used in thermal drilling systems that melt through the ice, is purified, UV treated, and filtered to remove any microbial cells before being directed down the borehole. Instruments that enter the borehole are scrupulously cleaned and decontaminated. The scientists refer to the standard of care required as analogous to what NASA applies when exploring other planets. You do not want to introduce surface life to an alien environment. The subglacial lakes of Antarctica, in terms of the care needed to approach them without causing damage, are treated as extraterrestrial environments. The first two subglacial lakes to be directly sampled were Lake Whillans in West Antarctica, accessed in January 2013 by the Wizard Project, Whillans Ice Stream Subglacial Access Research Drilling, and Mercer Subglacial Lake, accessed in December 2018 and January 2019 by the Salsa Project, Subglacial Antarctic Lakes Scientific Access. Lake Whillans, sampled first, yielded microbes. Living, metabolically active bacteria, tens of millions of cells per liter of lake water, thriving in the dark, sub-zero, nutrient-sparse environment. Their presence confirmed what many scientists had hoped but could not prove until that moment: that the subglacial lake environment, however extreme, was biologically active. The Lake Whillans microbes appeared to be feeding primarily on chemosynthesis, harnessing the energy in chemical reactions between dissolved minerals and gases, and on ancient organic carbon stored in the lake's sediments, carbon derived from biological material that had been deposited before the ice sheet covered the area. The discovery made enormous news in 2013, and for good reason. Confirming life in a subglacial lake, real, living, metabolically active organisms, had been a major scientific target for over a decade, and achieving it was a landmark. But the Whillans microbes, while significant, were exactly what the most optimistic predictions had suggested might be there. They were microbes. They were small. They were doing what microbes do in extreme environments around the world: surviving, chemosynthesizing, metabolizing at the edge of what biochemistry can sustain. Important, but not shocking. Lake Mercer, accessed 5 years later, was different. The Salsa Project drilled into Mercer's subglacial lake on December 26, 2018, boring a hole 60 centimeters wide and 1,087 meters deep through solid glacial ice using a hot water drill with a pencil-sized nozzle. The drilling fluid, the heated, purified water that melted the ice as the drill descended, was itself constantly filtered and UV sterilized during the process. A closed-loop decontamination system that represented the most rigorous clean access protocol in subglacial drilling history. Lake Mercer itself is not a deep lake. Its water column is only about 15 meters from the ice ceiling above to the sediment floor below, a shallow, flooded, dark room beneath a kilometer of ice. It covers roughly 160 square kilometers, an area twice the size of Manhattan, and it sits about 800 kilometers from the South Pole. Unlike Lake Vostok, which is thought to be a largely closed and stable system, Mercer is hydraulically active. It exchanges water with neighboring subglacial lakes through the subglacial drainage network, with water replacement times estimated at roughly a decade from the Ross Sea. It is, relative to Vostok, a young and dynamic system. The SALSA team extracted sediment cores from the lake bottom, including the longest sediment core ever retrieved from a subglacial lake. 5.5 feet of compressed lake bottom mud, representing thousands of years of accumulated history. They filled 6 10-liter bottles with lake water. They captured the first ever footage of the interior of a subglacial Antarctic lake, a camera descending through the borehole into the dark, narrow water column, illuminating 15 meters of water that had never been seen by human eyes. The water, as expected, was full of bacteria, tens of thousands of cells per milliliter, metabolically active, living chemosynthetic lives in the dark. But the mud was something else entirely. When project leader John Priscu's team put the sediment cores under the microscope at McMurdo Station, they found what they were looking for. And then they found things they were emphatically not looking for and initially refused to believe were there. Crustaceans. Not living crustaceans, but the well-preserved carcasses of crustaceans. Tiny shrimp-like organisms ranging from 0.1 to 1.5 millimeters in size, smaller than a poppy seed, but complete, intact, with all their structural details preserved in the mud. The shells of diatoms, photosynthetic microorganisms that require sunlight to grow, whose physical presence in the sediment of a lake that has been sealed under a kilometer of ice was initially impossible to reconcile with any known mechanism for how they could have gotten there. And a tardigrade. One tardigrade carcass, whole and recognizable, the characteristic barrel-shaped body, the 4 pairs of stubby legs, the circular mouth with its feeding apparatus. A water bear, as they are commonly called. An animal, not a microbe. An animal, in the sediment of a lake buried a kilometer beneath the Antarctic ice sheet. Priscu, by his own account, thought the finding was a mistake. He assumed contamination, that somehow, despite the rigorous decontamination protocols, a surface organism had been introduced into the sample. He had the team clean and sterilize their equipment again, from scratch, and resample. The new samples came back, and the crustacean shells were still there. Multiple shells, from multiple specimens, in multiple independently collected cores. The finding was real. The question of how those carcasses got there, how the bodies of crustaceans and a tardigrade, and the shells of sun-dependent diatoms, arrived in the sediment of a subglacial lake buried a kilometer under the Antarctic ice sheet, is one that researchers are still working to resolve. The most likely explanation involves the geological history of the region. The sediments of Lake Mercer sit in a basin on the floor of West Antarctica that, approximately 1 million years ago, was covered not by ice but by open ocean. The West Antarctic ice sheet has advanced and retreated multiple times over geological history in response to ice age cycles. At its minimum, it may have retreated far enough that the Ross Sea extended over what is now dry, or rather, ice-covered, land. The crustaceans and diatoms in the Mercer sediment may not have lived there under the ice. They may have lived there when it was ocean, and their remains were preserved in the marine sediment that accumulated over thousands of years during that open water period. When the ice advanced again, it rode over the top of those sediments and sealed them, and the crustaceans and tardigrade were entombed, perfectly preserved in the cold dark for 1 million years. Whether or not that explanation is correct, the discovery was significant for a reason that went beyond the biology of the individual fossils. It meant that the sediment record of subglacial lakes was a window into the paleoclimatic history of Antarctica, a record of what conditions had been like in the distant past, when ice had advanced and retreated and the continent had looked very different from its current form. Reading those sediments carefully, layer by layer, using the chemical and biological fingerprints they contain, was a way of reconstructing the history of an entire continent's climate. The most recent chapter in the subglacial lake story came in August 2025 when the Korea Polar Research Institute, in collaboration with American research teams including the SALSA scientists, published a landmark paper in the journal Nature Communications. The paper, titled Genetic Isolation and Metabolic Complexity of an Antarctic Subglacial Microbiome, reported the results of an extraordinary analytical effort. 1,374 single-cell amplified genomes obtained from individual bacterial and archaeal cells isolated from Mercer subglacial lake's water column and sediments. It was the world's first successful single-cell genomic analysis from a subglacial lake, and what it revealed was stunning in its implications. Most of the microorganisms in Mercer subglacial lake were not just unknown species. They were not just species new to science in the way that a newly described beetle or deep-sea worm is new to science, recognizably related to known lineages, just not previously cataloged. The Mercer microbes were genetically isolated from marine and surface biomes in ways that indicated deep, long-term evolutionary divergence. Their genomes were different enough from the closest known relatives that they likely represented entirely new taxonomic groups. Not just new species, but new genera, new families. Entities that split off from the known tree of life far enough in the past that their DNA had drifted into genuinely uncharted territory. Among the microbial groups found in Mercer was a significant representation of Pteridobacteria, a group of tiny bacteria, sometimes called CPR bacteria for candidate phyloradiation, that are characterized by ultra-small cell sizes, streamlined genomes, and unusual biochemistry. Pteridobacteria were only described as a major bacterial group in the 2010s, after the development of culture-independent genomic techniques made it possible to detect organisms that could not be grown in a laboratory. They are thought to be among the earliest diverging lineages of bacteria, representing a deep branch of the tree of life that was largely invisible to science until very recently. Finding them in abundance in Mercer subglacial lake, and finding that the Mercer versions of these organisms were genetically isolated from known Pteridobacteria in other environments, suggested that the subglacial lake had been a cradle of microbial evolution, a sealed refuge in which ancient bacterial lineages had continued to evolve in isolation from the surface world for thousands or tens of thousands of years. Microbes in the subglacial lake have evolved to survive without sunlight, in total darkness, and with extremely limited energy sources, said co-first author Dr. Kyun Hwang of the Korea Polar Research Institute. They display a range of metabolic strategies, oxidizing either organic matter or inorganic compounds depending on oxygen availability, and this metabolic flexibility is likely the key to their survival. That metabolic flexibility is worth dwelling on because it speaks directly to the broader question of what life is capable of in extreme environments. The Mercer microbes do not rely on a single biochemical strategy. When oxygen is available, from the slow chemical reactions between water and rock, from the traces of atmospheric gas dissolved in water that filtered down through the ice millions of years ago, some of them are aerobic, using oxygen in their respiration as surface organisms do. When oxygen is scarce or absent, others switch to anaerobic metabolism, oxidizing reduced compounds, methane, iron, sulfur, nitrogen. They have not been forced by their extreme environment into a single, narrow metabolic niche. They have, over thousands of years of isolation and evolution in a resource-limited environment, developed a community-level metabolic toolkit that covers nearly every available energy source, hedging against the uncertainty of a world where no single energy supply can be counted on indefinitely. This is the world's first successful single-cell genomic analysis from a subglacial lake, revealing microbial adaptation and evolution in darkness, low nutrient, and high-pressure environments, said President Hyung-Chul Shin of the Korea Polar Research Institute. Of more than 600 known Antarctic subglacial lakes, only 2 have yielded samples, making this a landmark achievement in polar science. Only 2. Only 2 of more than 600 known subglacial lakes have been directly sampled. The size of the unexplored territory is staggering. Lake Vostok, the largest and most celebrated of all the subglacial lakes, sits under 4 kilometers of ice, with its water estimated to have been isolated from the atmosphere for 15 to 25 million years, and it has never been directly sampled in a scientifically clean way. In February 2012, a Russian drilling team reached the water surface of Vostok after decades of drilling, their borehole finally punching through the last meters of ice into the lake. Lake water flooded up into the borehole and refroze, and that refrozen water was retrieved the following season. But the Russian drilling used kerosene and freon as drilling fluids, and the contamination questions that surrounded the resulting samples were significant enough that the biological findings, early reports of bacterial DNA, including what appeared to be sequences not matching any known organism, were not accepted by the broader scientific community. Lake Vostok, 15 to 25 million years of isolation, possibly some of the most ancient lake water on Earth, remains, in the strict scientific sense, unsampled. What is waiting in its water, what organisms, what biochemistry, what evolutionary history, is unknown. The speculation ranges from the conservative, microbial life similar in kind to what was found in Whillans and Mercer, but with longer evolutionary divergence, to the genuinely exotic. Some researchers have pointed out that the oxygen concentration in Lake Vostok's water is estimated to be extraordinarily high, perhaps 50 times higher than in normal surface lakes, a consequence of the enormous volume of glacial ice above dissolving oxygen-rich gas into the water over millennia. Such high oxygen concentrations would be toxic to most organisms. The organisms in Vostok, if they exist, would have to be adapted to an environment of excess oxygen, not a scarcity of it, as in most deep environments, but in abundance. What that kind of adaptation looks like, what biochemistry supports it, is entirely unknown. There is a recurring theme in the story of Antarctica's subglacial lakes that connects directly to the story of the ecosystem beneath the George VI ice shelf, and that connects both of them to something larger. It is this: Antarctica is not finished surprising us. It is not a static, fully mapped, well-understood continent whose basic contours of biology and geology we have reliably established. It is a place where the ratio of what is known to what is unknown is, even by the standards of our still young scientific knowledge of Earth, extraordinarily low. Every time we have found a way to look beneath the ice, whether with radar from aircraft or hot water drills bored through a kilometer of glacial ice, or an ROV descended from a research vessel into a sea cavity freshly exposed by an iceberg the size of Chicago, we have found life. Every single time. Life in a place we said life probably wasn't. Life in a place we said life couldn't sustain itself. Life that has been there for longer than we knew to look for it. Doing things we didn't know life could do in conditions we assumed were incompatible with anything we would recognize. The subglacial lakes are, in that sense, the deepest expression of the same truth that the A-84 discovery made visible. The Antarctic ice is not a barrier to life. It is a vault. And in vaults, things are preserved, protected from the disturbances of the surface world, from competition, from the restless reshuffling of ecosystems driven by climate change and geological upheaval and the ordinary processes of ecological succession. The things preserved in Antarctic vaults are not inert. They are alive. They are evolving. They are doing biology in the dark, in the cold, under the crushing weight of kilometers of ice, in complete ignorance of and indifference to the civilization that has spent the last century trying to understand them. What we have found so far is, almost certainly, a small fraction of what is there to be found, and what finding the rest of it will mean for our understanding of life's boundaries, for our models of Earth's history, for the possibility of life on other worlds, is where this story, ultimately, is heading. Section 6. The bigger picture. Everything in this video, every organism, Every discovery, every unanswered question, sits inside a larger context. And that context is changing, fast, in ways that make understanding what lies beneath Antarctic ice not just scientifically fascinating, but genuinely urgent. Not urgent in the way that scientists say things are urgent when they want funding. Urgent in the way that a ticking clock is urgent. Urgent in the way that a shore you can see from a sinking boat is urgent. Antarctica is melting. This is not a controversial statement. It is not a political position. It is a measured, documented, rigorously verified scientific observation made by thousands of researchers across dozens of nations over decades of fieldwork, satellite monitoring, oceanographic measurement, and ice core analysis. The Antarctic ice sheet, the continent-spanning mass of glacial ice that holds, locked in its volume, approximately 70% of the world's fresh water, is losing mass. It has been losing mass at an accelerating rate for at least 3 decades. The rate of ice loss from Antarctica has roughly tripled since the 1990s. And the consequences of that loss, for sea levels, for ocean circulation, for climate systems far from Antarctica itself, and for the extraordinary hidden ecosystems we have spent this video exploring, are among the most important and most difficult to predict phenomena in the Earth science of the next century. Understanding exactly what is at stake requires separating Antarctica into its parts, because not all Antarctic ice is the same, and not all Antarctic ice loss carries the same implications. The Antarctic ice sheet is divided by the transantarctic mountains running across the continent into two major components, the East Antarctic ice sheet and the West Antarctic ice sheet. The East Antarctic ice sheet is the larger of the two, far larger. It covers roughly 10 million square kilometers and contains enough ice to raise global sea levels by approximately 52 meters if it melted completely. For most of the period during which scientists have been monitoring it with satellites, the East Antarctic ice sheet appeared to be roughly in balance. Gaining mass from snowfall on its interior at roughly the same rate, it was losing mass from calving and basal melting at its margins. More recent data has complicated this picture, with some sectors of East Antarctica now clearly losing ice in ways that earlier models did not predict. And projections from a November 2025 study published in Nature Communications suggesting that under high emissions scenarios, the East Antarctic ice sheet could begin consistently contributing to sea level rise by 2030 and accelerate significantly by mid-century. But for the moment, the East Antarctic ice sheet, while not immune to warming, is not the part of the story that keeps glaciologists up at night. The West Antarctic ice sheet is. The West Antarctic ice sheet sits in a geological basin whose bedrock lies, in many places, well below sea level. This is critical. A land-based ice sheet, one sitting on bedrock above sea level, loses mass primarily through surface melting and iceberg calving at its margins. But a marine-based ice sheet, one whose bed is below sea level, is vulnerable to a different and potentially catastrophic mechanism called marine ice sheet instability. When warm ocean water intrudes under the floating ice shelves that fringe the West Antarctic ice sheet, it melts them from below. As the ice shelves thin and weaken, they provide less buttressing support to the glaciers flowing into them from land. The glaciers speed up. Their grounding lines, the points where they transition from resting on bedrock to floating, retreat deeper into the below sea level basin. And crucially, because the bedrock slopes downward toward the interior of the basin, a retreating grounding line encounters deeper and deeper water, which accelerates the melting, which accelerates the retreat. The process can become self-sustaining, a feedback loop that, once initiated, continues even if external warming stops. This is what researchers mean when they talk about the potential for irreversible collapse of the West Antarctic ice sheet. Not a sudden dramatic event. The ice sheet won't fall into the ocean over a weekend, but a centuries-long, self-perpetuating drawdown that, once started, cannot be halted by any realistic reduction in human greenhouse gas emissions because the physics of the feedback loop doesn't care what happens at the surface once the internal dynamics are established. A 2023 study published in Nature Climate Change, examining ocean warming projections in the Amundsen Sea, the region most critical to West Antarctic ice sheet stability, found that rapid ocean warming at approximately triple the historical rate was likely already committed over the 21st century regardless of emissions pathway. The study's conclusion was stark. The opportunity to preserve the West Antarctic ice sheet in its present state has probably passed. The West Antarctic ice sheet contains enough ice to raise global sea levels by approximately 5.3 meters, more than 17 feet. Not all of that would be released quickly, and the timescale for its full contribution is measured in centuries. But even a fraction of that contribution, delivered over the timescale of a century or two, would redraw the coastlines of every continent on Earth. It would submerge vast areas of Florida, Louisiana, Bangladesh, the Netherlands, Vietnam's Mekong Delta, and dozens of island nations in the Pacific and Indian Ocean that sit close enough to current sea level that a meter or two of rise is not a statistic. It is an extinction. A November 2025 study modeled Antarctica's contribution to sea level rise across different emissions scenarios and found that the highest regional rises from Antarctic melt alone, in a moderate emissions scenario, would reach up to 5 feet by 2200 in the Indian Ocean, Pacific, and Western Atlantic basins. Under a high emissions scenario, the average global rise from Antarctic melt alone would approach 10 feet by 2200. The highest impact regions are not, notably, the regions closest to Antarctica. Because of gravitational effects, the weakening of Antarctica's own gravitational pull on the surrounding ocean as it loses mass, sea level near Antarctica actually falls slightly even as Antarctica contributes to a rise elsewhere. The places that experience the most dramatic rise are far away: the Marshall Islands, Micronesia, Palau, the Caribbean. The people who will suffer most from the melting of Antarctic ice are people who live closest to the ocean, in the lowest lying parts of the world, in nations that have contributed least to the greenhouse gas emissions driving the warming. Within the West Antarctic ice sheet, one glacier above all others has come to symbolize the risks of what's happening. Its name is Thwaites. Scientists sometimes call it the Doomsday Glacier, though they tend to do so with the resigned ambivalence of researchers who know that dramatic nicknames are useful for public communication and uncomfortable for scientific precision. Thwaites Glacier drains a massive area of West Antarctica, roughly the size of Great Britain, and it is currently losing ice faster than at any point in the recent geological record. Its grounding line has already retreated significantly. The ice shelf that once provided some buttressing at its front has fractured, and the remaining ice is riddled with fractures that are expanding. Models suggest that if Thwaites collapses entirely, it could destabilize neighboring glaciers, potentially triggering a cascading retreat of the broader West Antarctic ice sheet. Thwaites alone contains enough ice to raise global sea levels by more than half a meter. As an initiator of the broader West Antarctic instability, its implications could eventually reach several meters. The waters beneath Thwaites Glacier, another sub-ice shelf environment, sealed, dark, unsampled, are among the most active in the cryospheric science community's research agenda. Understanding how warm circumpolar deep water is accessing the Thwaites grounding zone, how rapidly the ice is thinning from below, and what the seafloor topography looks like under the retreating grounding line are urgent scientific priorities. Every piece of knowledge about how sub-ice shelf environments work, including the knowledge generated by the January 2025 expedition beneath A84, contributes to that understanding. The George VI ice shelf itself, where A84 calved, is not merely a passive backdrop to the ecosystem story. It is a character in the climate story as well. Paleoclimate evidence from sediment cores around Alexander Island has established that the George VI ice shelf retreated significantly during a warm period approximately 9,500 years ago, during the Antarctic Holocene optimum, when summer insolation in the southern hemisphere was higher than today. At its minimum, the shelf may have disappeared entirely from what is now its northern section. It reformed over subsequent millennia as temperatures cooled. A 2025 study published in Nature Science examined ice shelf collapse events during the Holocene and found evidence of meltwater feedback loops that could dramatically accelerate the disintegration of floating ice. Mechanisms involving the pooling of meltwater in surface depressions that hydraulically fractures the ice from above, while warm ocean water attacks it from below simultaneously. The study found that collapse could occur much more rapidly than previously modeled once these feedbacks were engaged. The calving of A84 on January 13th, 2025 is, in isolation, a natural event. Ice shelves calve icebergs. They have always done so. It is part of the normal mass balance cycle of polar ice. But the Bellingshausen Sea has warmed faster than almost any other part of the Southern Ocean. Sea ice in the region has declined dramatically since 2016. The warming of the ocean beneath the George VI ice shelf has accelerated its basal melt rate. A84 is not simply a naturally occurring event that happened to provide a scientific opportunity. It is also a data point, one more piece of evidence in the accumulating record of a continent responding to a warming planet. Now set all of that aside. The sea levels, the glaciers, the coastlines, the political and humanitarian dimensions of climate change in Antarctica, and consider the discovery of January 2025 from a completely different angle. Consider it not as a climate story, or a conservation story, or even a story about the extraordinary resilience of life in extreme conditions. Consider it as a story about what life tells us about the universe. Jupiter's moon Europa is roughly the size of Earth's moon. It orbits Jupiter at a distance of about 670,000 kilometers, deep inside Jupiter's powerful radiation belts, far from the warmth of the sun. Its surface temperature averages around minus 160 degrees Celsius. Its surface is covered in a shell of water ice that appears to be somewhere between 15 and 25 kilometers thick, though estimates vary. And beneath that ice shell, maintained in liquid form by the intense tidal heating generated by the gravitational interaction between Europa, Jupiter, and the other Galilean moons, the constant flexing of Europa's interior creating frictional heat, like the warmth generated by repeatedly bending a metal paperclip, is an ocean of liquid water. Not a metaphorical ocean, not a geological inference from a distant line of spectroscopic evidence. A liquid water ocean containing more water than all of Earth's oceans combined. An ocean that may have been liquid for billions of years, longer than complex life has existed on Earth. An ocean whose surface periodically vents material upward through the ice in plumes that have been photographed by the Hubble Space Telescope, and that the upcoming NASA Europa Clipper mission, launched in October 2024, will investigate in detail as it conducts 49 close flybys of Europa beginning in the early 2030s. Europa has liquid water. It has chemical energy sources, interactions between water and rock at the ocean floor that could drive chemosynthesis. It has, by the criteria that astrobiologists use as their working definition of habitability, the basic requirements for life as we understand it. Saturn's moon Enceladus presents an even more tantalizing case. Enceladus is a small moon, only about 500 kilometers in diameter, small enough to fit within the borders of Texas, but it has, in the language of planetary science, demonstrated its ocean to the universe in the most dramatic possible way. It is actively venting it into space. From the south polar region of Enceladus, jets of water vapor, ice particles, and organic compounds spray outward through cracks in the ice shell, the tiger stripes, as they are called, forming a vast plume that the Cassini spacecraft flew through multiple times during its mission. Cassini's instruments detected water vapor, ice, salt, silica nanoparticles, and complex organic molecules in those plumes. It detected molecular hydrogen, a chemical signature that, on Earth, is associated with the hydrothermal reaction called serpentinization, in which water interacts with certain types of rock to produce hydrogen gas that can be metabolized by certain microorganisms. The detection of hydrogen in Enceladus's plumes was not, by itself, proof of life, but it was evidence of exactly the kind of chemistry that, on Earth, supports chemosynthetic ecosystems at hydrothermal vents. The question of whether life exists in the oceans of Europa and Enceladus is, right now, unanswerable. We have not sampled those oceans. We have no way to sample them with any currently operational mission. The closest humanity will get in the near future is the Europa Clippers flybys and the long-discussed, never-funded Europa lander concept, or the ESA mission to Enceladus, whose preliminary planning targets a launch in the early 2040s. But here is where Antarctica becomes not just a place to study for its own sake, but a proving ground for the science we will need to do elsewhere. Every technique, every instrument, every methodological approach, every lesson learned about how to access a sealed, ice-covered liquid water environment without contaminating it, without destroying it, while gathering meaningful biological data from it. All of that is being developed, tested, and refined in Antarctica. The hot water drills that bored into Lake Whillans and Lake Mercer. The decontamination protocols developed over decades of drilling programs. The ROV technology that sent Sebastian into the darkness beneath A-84. The glider technology that measured meltwater flows and biological productivity in the subshelf cavity. The acoustic modems that allowed communication between underwater instruments and surface operators. All of it is the infrastructure of a planetary-scale scientific capability being built in the most inaccessible place on Earth as preparation for eventually using it in the most inaccessible places in the solar system. Lake Vostok, isolated under 4 kilometers of ice for possibly 25 million years, is the closest thing on Earth to the ocean of Europa in terms of its isolation, its darkness, its pressure, and the potential long-term evolution of whatever life exists within it. The subglacial lakes of West Antarctica, hydraulically connected, dynamically active, their water cycling through the system on timescales of decades. These are analogs for the more dynamic ocean worlds, where circulation and exchange between water and ice and rock are ongoing. The ecosystem beneath the George VI ice shelf, complex, ancient, sustained by mechanisms we do not yet fully understand, thriving in conditions we had said could not support it, is a standing demonstration that liquid water under ice, given enough time and enough geological context, produces life. Not microbial mats clinging desperately to the edge of existence. Not a handful of cells per liter of water. Life. Full ecological communities, centuries old, with food webs and predators and foundation species and structure. If that is what liquid water under ice produces here, on Earth, in a geological and oceanographic context that is in many ways less favorable than what models suggest exists beneath the ice of Europa and Enceladus, what does that imply about what those oceans might contain? The honest scientific answer is, we don't know, and we won't know until we can send something there to look. But Antarctica has shifted the frame of the question. Before the 1977 discovery of hydrothermal vent ecosystems, the question of whether life could exist in those oceans was answered tentatively, hypothetically, by inference and analogy. After 1977, it was answered in principle. Yes, life can exist without sunlight, without surface connection, driven by chemical energy in the dark. Antarctica, over the last decade and especially since January 2025, has extended that answer considerably further. Yes, not just microbial life, not just the minimal expression of biology at the edge of what is thermodynamically possible, but complex, differentiated, multi-level ecological communities. Filter feeders, predators, mobile organisms, and sessile ones. Ancient lineages and dynamic relationships. Hundreds of years of uninterrupted biological history in the dark, under the ice. There is one more dimension to the bigger picture that is not about climate or astrobiology, but about something more fundamental, about what kind of species we are and what kind of relationship we are capable of having with the planet we inhabit. The history of human exploration has, for most of its length, been a history of exploitation. We went to places, we extracted what was valuable, we moved on. We went to Antarctica and we killed penguins for their oil and seals for their fur and whales for everything, and we left the bones on the beaches, and the beaches are still full of bones, still, right now. If you walk the right stretches of South Georgia Island or the South Shetlands. We charted coastlines in order to find resources. We drilled ice cores in order to understand past climate primarily, because understanding past climate tells us something about future conditions, and future conditions determine the economic and strategic calculus of nations. Even our scientific expeditions, as disinterested and pure in their intentions as the individual scientists on them genuinely are, exist within a framework of resource calculation that shapes which questions get asked and which get funded. But something about Antarctica resists that framework. Something about the scale of it, the scale of the cold, the scale of the distance, the scale of the silence, makes the ordinary logic of exploitation feel inadequate. And something about the discovery of life in its most hidden, most inaccessible, most improbable corners. Centuries-old sponges under a floating ice shelf, microbes in a lake sealed under a kilometer of ice for thousands of years, giant phantom jellyfish drifting through a darkness that had never been broken by human light before January 25th, 2025, makes it impossible not to feel that what is at stake here is something larger than a scientific result. What is at stake is the possibility of life. Not metaphorically, literally. The West Antarctic ice sheet is under threat from warming that is, at this point in the scientific assessment, committed over the coming century regardless of our near-term emissions choices. The ice shelves that create the habitat conditions for the ecosystems beneath them, the floating ice that seals the dark cavities, maintains the temperature and pressure regimes, drives the meltwater circulation that apparently feeds the sponges and the corals and the ice fish. Those ice shelves are thinning, retreating. Some have already collapsed entirely. The Larsen B ice shelf, on the eastern side of the Antarctic Peninsula, disintegrated over the course of a single austral summer in 2002, losing 3,250 square kilometers of ice in 35 days. What was under it has never been properly studied. Whatever centuries of biological history it had been protecting was exposed, destabilized, and in many cases lost before science even knew it needed to look. The George VI ice shelf is thought to be more stable than Larsen B was in 2002. But the Bellingshausen Sea is warming. The ocean beneath the shelf is warming. Models of the shelf's future under continued warming scenarios are not uniformly optimistic. And the ecosystem that Sue Bastian documented in January 2025, the one that has been growing in the dark for what is probably hundreds of years, has no particular resilience against the introduction of warm ocean water, or against the destabilization of the ice overhead, or against the competition from faster-growing organisms that will colonize from the open ocean as the conditions change. The sponge that has spent a century growing 2 centimeters per year in water that hovers just above freezing has not evolved the capacity to adapt quickly. It has evolved the capacity to endure. And endurance, in a world where the conditions of endurance are changing faster than evolution can track, is sometimes not enough. Dr. Eskeet said it clearly: we need to come back. We need to keep studying. We need to understand what drives that ecosystem, because without understanding it, we cannot know what losing it would mean, and we cannot know what we would need to protect to preserve it. The monitoring instrumentation, the gliders, the moored sensors, the acoustic equipment, that would give us that understanding does not install itself. It requires funding, planning, international cooperation, and the kind of sustained institutional commitment that is hard to maintain in a world where the scientific funding landscape shifts with political cycles and the attention of the public is always drawn to the next extraordinary thing. The next extraordinary thing, in this case, might well be beneath another ice shelf, or in another subglacial lake, or in a place we haven't yet thought to look, in a darkness that has been growing for longer than we have been curious about it. Here is what we know, as of early 2026, as a result of everything this video has described. We know that the Antarctic ice hides ecosystems that we did not know existed. We know that life, given liquid water and time and the quiet persistence of geology, will find a way to organize itself into something more complex and more beautiful than any model or theory predicted. We know that the mechanisms that sustain that life in total darkness are not fully understood, that the science of how organic energy moves under a floating ice shelf is still being assembled from the data of one extraordinary expedition and the gliders it left behind. We know that subglacial lakes hundreds of kilometers inland hold microbial communities whose genomes represent evolutionary lineages that split from the known tree of life thousands of years ago. We know that the organisms in those lakes have developed metabolic strategies so flexible and so diverse that they can survive on nearly any available chemical energy source, hedging against the uncertainty of a world that offers very little. And we know that what Antarctica has shown us about life on Earth, that it can thrive anywhere that liquid water and energy persist, regardless of whether sunlight reaches, regardless of whether the surface world knows the ecosystem is there, has direct and profound implications for the question that may be the most important one science will ever ask. Are we alone? We are not alone on our own planet. That, at minimum, January 25th, 2025 established beyond any reasonable doubt. The ocean beneath A84's former territory was not empty. It was not marginal. It was not a thin rind of life clinging to the edge of survivable conditions in the hope that something better would come along. It was alive, completely and abundantly alive, in the dark, under the ice, for centuries without us. How many other places on Earth are like that? How many other places in the solar system? How many other places in the universe, around other stars, where ice covers liquid water and geology provides energy and time does what time always does, eventually, which is produce life where the conditions allow it? We don't know the answer to those questions. We may not know for a very long time. But the shape of the answer has changed permanently because of a slab of ice the size of Chicago that broke away from a floating shelf on January 13th, 2025, and because of a team of scientists who had 12 days to make a decision and did not hesitate. The ice broke, the lights came on, and the world was larger than it had been the day before.